excellent early session of constructives, and now we're going to have rebuttals from each member. Eric and I have decided to extend this debate by 30 minutes under the rules, so each speaker will have an hour and 15 minutes for these rebuttals, and Yuri will start us off. So please go ahead, Yuri.
All right. Yes, so basically I have some slides of Peter's that I just want to make some comments on, and the structure is just I'll show Peter's slide, and then I'll maybe go back to my note and either go back to the slide or just we'll speak. And basically, yeah, so the slide is that, the lab is not some sort of like, I don't know, military institution with a secret, like a fortress full of deadly viruses, but just a bunch of research scientists. And of course, I mean, yes, the same labs that have leaked viruses before look like this. So like the Taiwan BSL-3 lab and all the other labs in China that leaked SARS-1. So I don't see how this, is relevant to what we're trying to establish. Whether
am I allowed to interject at any time, or is all him right now?
I think we should do the 15 minutes
okay, cool.
of each person and then interject afterwards. Okay with you, Eric?
Yeah, can we at least save the interject? We'll get more free form as time goes on, but for now, let's give each of them some time.
Okay, yeah.
Not bad.
So, right, and I guess this is the next slide I wanted to address. Basically, yeah, the estimate to why exactly they only had 200 sarbecoviruses. I mean, they did mention in diffuse that they had over 180 samples, and Peter says that they published them all, but of course, I think it's pretty standard to work on unpublished viruses. I mean, that's the whole kind of sequence of events during research. You find something, some novel, interesting virus, you do novel, interesting work, and then you publish. And of course, those papers that Peter referenced, that some of them were published in 2018 or 2019, or some viruses were like put on GenBank to be embargoed, with an embargo to be released in like 2020. But of course, that's one paper that was already kind of written, like the manuscript, but there is very likely to be a lot of research that has yet to be published and that's still being worked on. So to say it's a secret virus kind of presupposes some sort of malfeasance, but like research, you have a lot of ongoing projects that you don't publish anything, you don't even mention anything because you're afraid of getting scooped by your frenemies, like Ralph Barrick's group, I'm sure, was also interested in a lot of the things that WIB was working on. So I'm sure they had a lot of unpublished viruses. And I mean, we know they had a lot of unpublished viruses just based on the stuff that Peter presented, like 200 viruses, and they published them eventually. But of course, I think it's absolutely plausible they had more viruses they didn't yet get to publish. And in terms of like how much gain of function work they have done, I think like what you classify as a gain of function, and just because they published many more papers than just actual gain of function papers, with like some reviewed papers and some other papers that did not have like gain of function research, I don't think you can somehow like use that proportion to say that it's very unlikely they did not do any novel gain of function research, because obviously we know that not only did they have the diffuse proposal, which was not funded, they actually have a grant. They had a grant, active grant, that was funded by the NIH, which was stopped right at the pandemic, with Wuhan Institute with EcoHealth, and it involved creating even more novel chimeras along the lines of the 2017 paper, where exactly they had 8 different chimeric viruses, where they inserted novel spikes into this backbone. And of course, the backbone, I mean, I think we can split hairs, but the backbone is the whole sequence or just missing without the spike. I usually think when they say reverse genetics backbone, they mean like a virus, the whole sequence of a virus, where you can mix and match or like just change, genetically modify parts of the virus. But it's a very minor point. But so in terms of this particular slide that says that diffuse could not have resulted in SARS-2 because they would have used known backbones, but I think I already addressed this point that these backbones were selected as a first step. These like WIV1 or WIV16 or SHCO14, these were known viruses, and they used multiple backbones, and they even mentioned this in the fact that in the diffuse grant that they wanted to use different backbones because that increases the chances of either isolating the virus or maybe even because some different parts of the backbones have different genes that can affect the potential to infect human cells. So, but the main point is that they plan to validate findings with the chimeric viruses that use known backbones. They plan to validate these findings with the original virus. The original, I guess, they would create backbone for new reverse genetic systems based on the original strain. And they also mentioned that they were planning to generate 3 to 5 novel backbones per year, even outside of the context of the, like, whether they found interesting results or did not find interesting results, but some spikes. So in terms of, by now we know it's not RATG13, very unlikely to be because we have, first of all, closer viruses to SARS-CoV-2 with the same receptor binding domain. And so it would have to be, the backbone they would have used has to be something that have not been published. If you want to call it a secret virus, it's fine, but I think it's misleading because I'm sure they had a lot of unpublished viruses. And in terms of RATG13, I don't want to get into these because obviously the known viruses that we have right now are too distinct on the nucleotide level from SARS-CoV-2 to be the exact backbone. Obviously on the protein level, banal 52 is like 99% similar, but of course on the nucleotide level it's 97% similar. So most likely they would have, if they did use a natural virus for the progenitor of SARS-CoV-2, it would have been closer to SARS-CoV-2 on the nucleotide level than whatever we know right now. RATG13, like this point, it's pretty much an irrelevant point, although early on, yes, it did make people suspicious, including myself. So in terms of the WIV not noticing the furin cleavage site and cutting off this diagram, this alignment right before the furin cleavage site, it's not just me that thinks it's suspicious. Other people thought it's suspicious, including virologists like Stuart Neal, who said he actually asked WIV why did they not mention the furin cleavage site, and they said they just didn't notice it. And it was 2 papers co-authored by Shizheng Li, and the first one was co-authored by her and Shibo Jiang that I mentioned before, who previously created a furin cleavage site that included the CGG codon. So Shibo Jiang is not just somebody that knew Shizheng Li, they cooperated on several projects, and they co-authored the paper on SARS-CoV-2 about where, and they mentioned where it's cleaved at the S1, S2 junction, like the protein number, but they didn't mention that it's actually now cleaved by furin as well. It wasn't just me who was suspicious. People were suspicious. Yes, I was incorrect. I thought it's an artifact cut off by hand, but it's actually not. I made that correction. I publicized that I was incorrect later on. I talked about this renaming with Brett Weinstein. This was like a June 2020 video, which is essentially just like a few months after the outbreak, and it was very early on. And the Wuhan in virology was very kind of suspicious in how they handled the RATG13, RA4991. They even implied that they sequenced RATG13 only after the outbreak, and we had to essentially kind of catch their hand when they released the sequencing data. We looked at it and saw that it had 2017, 2018 dates in the amplicon files, and we were very surprised because everybody thought at the time that our RATG13 was freshly sequenced. So WIV had to then confirm that no, we sequenced it actually years before the pandemic. And this clip that Peter played of me thinking that, one hypothesis was that actually the two are different, because in one of the early papers that we later uncovered, like a master's thesis that used RA4991 and fully sequenced it, they had a table of its similarity to SARS-1, including its spike gene similarity, and the percentage of similarity to SARS-1 they had listed for RA4991 was different from what...
5 minutes of the first 15 left.
Oh, wow. So basically, that was the hypothesis why the two might be different, and later on it turned out that they just used a different alignment algorithm or a different algorithm to calculate the percentage difference, and if you use that algorithm, the RATG13 and RA491 are the same virus. So, but of course, by now I think RATG13 is no longer relevant. Do they have undisclosed viruses closer than RATG13? Just because EcoHealth published all those viruses doesn't mean they disclose everything they had in their collection, because I'm sure they have active research right now and they wouldn't disclose active research. Oh, and by the way, in terms of gain of function research, they had, not only did they have that ongoing grant with EcoHealth, they had another ongoing grant with EcoHealth for MERS-like viruses, and they generated a bunch of MERS-like chimeras, which we wouldn't have known about unless we, because people FOIA'd their NIH progress reports. So in terms of whatever, the published viruses, I think it's not relevant. This basically short window between August 19 and 2019 when they could have sampled the secret virus, I think it's not correct. I'm sure they could have sampled it. I'm sure they had more viruses that they've been working on for years outside of whatever was published in conjunction with EcoHealth in the Latin paper. Daszak, I'm sure Daszak does not know everything that WIV had in progress, and I'm sure he did not know about the lab leak, but of course I'm sure he would be defending his collaborators while he has plausible deniability, like he wouldn't know if they had a lab leak at all. I won't get into accelerating mutations, although you could, and you'd see a pattern of mutations that is actually similar to the pattern between RATG13 and SARS-2. Basically it's making the same point that you closely guard your research before publication because you don't want to get scooped. Rare, coronaviruses do have furin cleavage sites, but in SARS-like viruses, SARS-2 is the only one. In terms of, one mutation away from an FCS, a whole bunch of viruses are just one mutation away from an FCS, like RM102 is just one mutation away from an FCS, and Banal 116 and 247 are also just one mutation away because it's the same fragment that they have. Two places, S1, S2 is the important junction, and in MERS they actually they showed that this is the cleavage site in this junction that, like in the words of Barak, unlocks the barrier to human emergence. 12 letters in terms of, in frame, it's obviously in frame, but it's just because if it was out of frame, you'd have a frame shift and it would be, most likely it's just also SNV mutation just before the insertion, like this TCT versus TCA in other viruses. So in terms of the insertion, people claiming that PAA is an insertion are completely wrong. We've shown this in 2021. We've shown this subsequently, not just ourselves, but many other now alignments exist because of many more viruses. Because the insertion claim was made only when RM102 was the only virus that had this fragment, no known virus that had this fragment. But of course, later on, you'd have banal viruses, two of them. You have the Cambodian viruses, RACCS, three of them that have this fragment. And to think that all of them have the same insertion in parallel with the deletion, it's nonsense. And any alignment algorithms that you use these days they don't produce this as this kind of sequence of deletion followed by an insertion. They just align it with the existing nucleotides, showing that it's most likely a point mutation that led to PAA. And it's actually an important point I wanted to make. Peter's saying that it's possible that this could have in nature turned into PRRA, and while it's possible that in these viruses this turned into PRRA, it's not possible that it turned into PRRA or became an insertion in the Wuhan virus, because the flanking sequences, which unfortunately on this slide you can't see, but actually here you see, the flanking sequences are very different between the PAA containing strains and basically the insertion, whether you have to insert this, including the QT fragment that you would need for this to be well cleaved. But so basically, if this evolved in nature, you'd see this in these kind of strains like RM102. But all strains that have PAA or PVA as this fragment, they don't even have the same receptor binding domain as the strains that contain this QT fragment.
We just wanted to pause you here, Yuri.
Yeah.
That's the first 16 minutes, I think. You can go through any of these slides afterwards as well. I think we just wanted to get a flavor of that start. Time is up to you, but I think it would be nice to, if those are your main points you wanted to start with. I don't know, Eric, if you wanted to.
I mean, if it's fine with Peter, I'd be happy to have Yuri keep going. Of course, he'd be using his time.
I think this is hard because I'm just writing objection after objection, and it's not really much of a debate if he says 30 things and I try to remember, and you've got a bunch of your own. Yeah. So why don't we actually talk to each other a little bit?
Yeah, all right. If it's okay, shall we switch it over for 15 minutes?
Or, we back it up to the slide you're just looking at with the QT.
Can I ask, I'd like to start you go, Peter, and then I'll ask clarifying questions.
Okay, yeah.
All right.
Yeah, everyone can talk. I mean, so when we sequenced we passaged the virus in Vero cells, QT was a common deletion, or I think in that case it was QTQTN. So the general idea that you can have a characteristic deletion that happens over and over and over, repeatable in experiments, can be like that is perfectly plausible. So you could have something in nature that has, originally one version has like the QTQT and also that insert, and that recombines or mutates. Also, you also said this is one nucleotide mutation away from COVID, or no, from having an FCS. So that's inconsistent. Which one nucleotide would you change here to make it an FCS?
Yeah, the P to an R could mutate obviously.
You need to add an R though. That's adding 3.
No, the P itself. You'd have, instead of PAAR, you have RAR.
Oh, okay, and you change one there, and you'd have a pair. Well, but they did an experiment saying RAAR isn't good enough for COVID. You need two leading Rs. But yeah, close enough. But the other thing I would point out here is it's not close enough for evolution, but it's also not close enough for the lab. So your lab leak theory is saying, a person would totally look at this and turn it into COVID, but that person would also have to add the QT, and they'd have to make the flanking sequence good enough. And you're imagining that person would just happen to see the PAA and say, that would be a perfect virus if I just change this. And you can't do that unless you've seen COVID already. There's no way to foresee that.
Well, I would say that if they saw this fragment, PAAR, and they were modeling its evolution into a furin cleavage site, they would then insert that potential furin cleavage site into like their high-risk strains or low-risk strains to turn them into high-risk strains like they were talking in diffuse.
But they wouldn't even see an insertion in your theory. You said it's not an insertion. So they do the genome comparison. They do the alignment, and it wouldn't look like an insertion, but they'd suddenly see that it was valuable as a cleavage site?
No, they wouldn't see this. I mean, this is not an insertion in the original strain, but they can model, like, for example, this getting inserted or getting recombined with QT carrying strain. And it's interesting that they're like within the Banal strains, like Banal 52 has QT, it has the receptor binding domain, but it does not have the PA fragment. But Banal 116 and Banal 247 they have the PAA fragment and they don't have the receptor binding domain, they don't have the QTQT. So basically someone looking at these samples could be modeling the very evolution that you claim occurred naturally, right? You claim that this somehow naturally turned into PRRA and then somehow recombined with the progenitor of SARS-CoV-2. And this is exactly what they could have been modeling, and they could have been modeling this in like this lazy way of okay, let's just insert the PRRA into SARS-CoV-2 and see if it makes a difference.
But you're saying it's not an insertion, and you're saying that if you run an alignment, it wouldn't look like an insertion.
It's
so the
an
person
insertion
they'd see a mutation, and then they'd have to say, what if I imagined this mutation was actually not a mutation and it was an insertion? No one would think that. Your theory is inconsistent with the two things you're saying here. You either have to pick whether it's an insertion or whether a person would understand this. You can't have both.
It's not an insertion in the original strains. It's not an insertion in RM1YNO2. It's not an insertion in Banal 16. But a genetic engineer could model this getting into SARS-CoV-2 progenitor by means of an insertion. So they're like, okay, what if in nature a virus like RM1YNO2 that evolved this furin cleavage site from PAA to PRRAR met a Banal 52 virus that does not have a furin cleavage site, and then they recombined and this got inserted into the Banal 52, or this created a furin cleavage site. And the kind of the easiest way to model this by genetic engineering is just, plumb the PRA, the 12 nucleotides into Banal 52, and this is how you get your furin cleavage site.
But what's special about the PAA? There's no R's in it, so nothing about it indicates that even if you turned it into insertion, it would be a good cleavage site insertion. How is that different than like any other one on this list? The next one has SIL. Why not mutate? Why not insert that after the QTQT and then turn that into a cleavage site?
I mean,
why not put RK into any of these?
I mean, any SARS-like strain can be argued that it can be turned into a furin cleavage site, and they could then, use that as an insertion. And they could have had several candidates based on whatever kind of viral families they were sampling. They're talking about this quasi-species with like within 5% difference of each other there, which is actually, by the way, I think you're misreading the diffuse proposal saying they were only looking at strains that are 5% away from SARS. I think they were looking at basically like low.
Pull that up in a minute, and we'll actually look at the words, and then we can.
Okay, so yeah, so basically, yeah, I'm proposing that they could have been looking at these some proto cleavage sites in these quasi-species, basically viruses that are circulating as this kind of cloud of mutations, and then whatever they said, high-risk strains and low-risk strains, and low-risk strains basically at low abundance might have some sort of proto-cleavage sites which would then they would engineer into these high-risk strains and create cleavage sites. So they could have a panel of potential cleavage sites, and the PRRA could have been just one of them, inspired by, maybe they saw a whole bunch of viruses like Banal 116, Banal 247 within their, cave that they were working on, or a particular bat species that they were looking at, and they were modeling how those could turn into cleavage sites and then get inserted into other core circulating viruses with a receptor binding domain that has high affinity to the human receptor.
Maybe. I mean, there's just so many combinations there between, QT and possible inserts, and you could find if they have 180 viruses here, you're going to have some combinatorial amount there, and I don't think this one's going to show up as meaningful until you've seen COVID.
Well, I think if we look at just the Banal sequences, it's interesting because they're subdivided into two categories. There's one kind of subclade with QT, and a lot of them have, not a lot of them, two of them have, or three, same receptor binding domain, Banal 236, Banal 52, or actually, they have very similar receptor binding domain as SARS-CoV-2, and like the second subclade has PAA, different receptor binding domain, and no QTQT. So you like, it's almost as if you'd want to like experiment and see if there could be potential recombination to arrive at.
Except the Banel viruses were found in 2021, so you have to hypothesize that they had those viruses in the first place, that they secretly went to Laos and found the same thing.
2020, actually, I think, but.
Can I interrupt?
Yeah.
I mean, unless you want to keep talking, I won't stop you then. I sort of feel like the speculative question of like, what idea could a researcher have had given the information they had at the time? Which this is a relevant question, but I think it's also a little bit unfalsifiable. We can never say, "Oh, you would never think of that," because someone could. I think more interesting but very similar question is the question of can it be natural? The question of could this 12 nucleotide insertion have arisen naturally? I think that half of the question is something I think we're much more likely to make progress on discussion. So if you want, I would suggest we steer the question in that direction.
That's actually harder for me to answer because I can't imagine what evolution would do.
Anything can be
about what humans, right? And
it's just very.
And, you can read the Slack messages with Christian Anderson and everything, and they found this PAA, and they're like, is that a mutation? Is that an insertion? But that was one of the reasons they changed their mind. They found the pangolin viruses, and they found this. And you can say they're bad faith actors, but from me, this is an update. Oh, if you can find something similar in nature, maybe it's more likely that it's found in nature. But I think you maybe have like a more conspiratorial version of thinking where like once you find this, you think, oh, well, what if the lab knew about this? And like I think I gave you a lot of trouble about like your early RATG13 theories. It's misnamed, but I guess the point there is not to like say that you've changed your mind. But if you were that suspicious about all these other things, about RATG13 being renamed and these 8 other viruses they were hiding, and none of that panned out as important, should you maybe downgrade your priors about these other suspicions you have about they have hundreds of secret viruses and secret sampling trips, and they saw this PAA virus?
Well, I don't see how the two are connected, because I mean we know they had hundreds of sampling trips and they have had thousands of samples, and to think that,
but not secret ones. Like,
well, not yeah.
You're saying the 180 viruses, and we saw the 2018 paper, and that had like 163, and then we saw the 2019 paper that had 200. So, like the Occam's razor explanation is the 180 is the same number, and it's just the viruses they had. Whereas your brain seems to go, well, what about the secret viruses they also had?
Can I?
180 is by 2018 they were publishing this in Diffuse. So since between 2018 they
the
did
secret
many
version
more
is the same as
trips.
the public version.
Okay, wait. Let me pause things because I want to establish things. We've just gone through a tremendous number of things. Let me try to resolve 3 things, or just get clarity on 3 things. So the first thing I want to start with is just understanding the objection about alignments, insertions, QT sites, the QT site, etc. I hear Yuri saying there's potential for mixing and matching here. There's a priori you could look at a wide range of existing viruses that you have, whether published or not, and you could see some pieces that you'd like to mix or match, and you might mix or match those in different backbones. The fact that there was a natural origin of them does not mean that you wouldn't insert it somewhere else. Is that a fair description of what you're saying about the various sites here, Yuri?
Yeah, definitely the mix and matching part. But also, yeah, this is not an insertion, and like when I guess Eric was asking, could this have arisen naturally? This is not how it could have arisen naturally. Like, this would not have resulted in this 12 nucleotide insertion.
Right. Okay.
You're saying that the PAA we're looking at in this slide could not eventually yield PRRA. That's what you're saying.
I'm saying it could not yield a PRRA insertion of 12 nucleotides into SARS-2 progenitor. I mean, that would be like an astronomical coincidence, because all of the flanking sites at each side are different. Like,
okay, Peter, so now what I hear you saying is that combinatorially it is unreasonable to expect one, that they would have identified all of these different sites as important because they don't know what they are a priori, given all of the kind of questions of differences from SARS. And two, if they were going to make those, they one, they wouldn't have had any a priori reason to do so, and two, it's astronomically large number of combinations, so why would it have resulted in that? Is that a fair description?
Yeah, that's about right. And you can add RRK to any of these viruses, and it's got a good cleavage site. And maybe I can imagine the case where maybe they found the good RBD if they had a secret virus and they said, here, this receptor binding domain is really great for humans, and they had a really good screening mechanism. I can see why they maybe start there and then add a good cleavage site to it. But the part where they spot PRRAR as important based on this partial alignment, I don't see how they would do that when you haven't seen COVID before. And also, Yuri is correct that anytime you align two sequences, it's arbitrary what you call a deletion, a mutation, whatever. I think the QT deletion is something that happens. We have evidence of that. So I think it is possible there's a natural one that has like both of these sequences, and that one, if it does exist, could mutate into COVID. But that's speculation, right? I don't know what nature has to work with.
You said the QT deletion is a natural thing. So do we have something that has in nature that has neither the QT nor the PAA or something similar in that spot?
I mean, there's ones on the list here that don't have either of those features, but
or have a deletion that have. So as I read it, all of the viruses we're looking at now, except for SARS-CoV-2, have the same number of nucleotides in this section. I feel like this.
That's just an artifact of the alignment.
Yeah, I don't know if that's an important point. If that's. I don't know.
Well, I feel like this is important to what Yuri is saying. Yeah. Yuri is saying that it's not a deletion followed by an insertion, and his argument is, in part, that the number of amino acids is conserved, and you sort of expect a deletion followed by insertion.
If you look at a wider family of sarbecoviruses, it's not conserved. Like if you look at that one I talked about with RAKQ that was found by Tom Peacock, that one doesn't have the same number here. So to some degree, the similarity in nucleotides is an artifact of you've already picked out the most similar ones to do your alignment here. And I think
Can
if
you
you
bring up an example on the screen? Just
we
talked a lot of viruses.
Yeah,
I think in general, having a local alignment. I presume this was generated from a local alignment on just S. Is that true?
I mean,
probably.
There's just. I think
pretty well.
I mean, this is Vincent Rancinello's slide, I think, so I don't know how he filtered this.
But actually, it's important. The length of this fragment is important, and all of the...
yield the screen, and I'll show a different version one.
Okay. Well, I hope you're having a good time running the timing over there.
We're operating under each of you is losing several seconds each time, and it will even out. So doing the best I can. Call me out if I'm screwing it up.
Can you see that? Yeah. So this is the RAKQ one that's almost, and then they've changed it into RAKR in their.
Can you just tell me what these viruses we're looking at are?
SARS-2 is what we all get infected with. Delta CS, I think that's a modified version where they must have removed. Oh, I think that's a deletion in culture where it lost the N through S. And then this is a virus, a sarbecovirus I think they found in Europe actually, which has almost.
WT here means wild type, right?
Wild type, yeah, sorry. And then the Q is they've mutated the Q to an R at that position. So the length there is not conserved. Instead of a QT, I guess they have an NT. I could find the bigger, but then they don't have the flanking there. And if you did this for coronaviruses across a bunch of species or even more sarbecoviruses, you're going to see a variety of lengths here. It's really just an artifact of we picked a certain number that are similar to SARS.
But actually, it's an artifact of the bats and the ecosystem in which we're looking for these sarbecoviruses. Because UK bats, it's a wildly different ecosystem. They have wildly different viruses. if we're talking about like SARS-2 like viruses, they have, it's actually very conserved that they have this length of this fragment at this S1, S2 cleavage, and the RMYNO2 and banana 116 ones that have the PA fragment are all 2 amino acids shorter at this junction actually. And to answer your question, Eric, no, we do not have any strains that are missing QTQT.
Wait, they're shorter. They're not actually, once you align them, they're not the same. So then that's not conserved.
No, I mean, in terms of the seeing similarity to like the UK viruses, right?
No, but you said the
Wildly different.
QTQT is removed, so that's 4 deleted, and then you said the PAA is added. So
No,
four
PA
minus
is like
3
a different. It forms a different subclade that's actually 2 amino acids shorter. Yeah, like
Yeah, so
all
it's
of
not
the
conserved.
QTQT
Too shorter.
containing. I don't see. I don't know anybody who is shorter at this.
Though you just said they're shorter in that clade. They're not the
They
same
don't
as
have
the other.
QTQT. No.
Because, but 4 have been removed and 3 have been added. So we're just, so there's no rule of nature that says the total number there is the same.
I mean, it's not a rule of nature. It's about this kind of this S1, S2 loop being kind of the right length, and I think you're pointing out this QTQT elongates it and it makes it accessible to cleavage, and so this length is actually an important.
So the 4 are gone and 3 are added. So it's not the same length as the other ones, is all I'm saying there. Sorry. Keep talking. Yeah.
Right, unlike SARS-2, which actually has an additional 4 added and elongates that loop to something we've never seen before. Or
Yeah.
since,
So it can be 0 or +1, or it can be +4. I mean,
but we
I don't
haven't
know.
seen any viruses like these. Oh, you can't see my cursor because I'm not sharing the screen. We haven't seen any viruses with like PAA that have, say, just the deletion.
So, but imagine we found one of those. Would that change your opinion on lab leak, or would you just say, "Oh, that means they must have had one of those, and so it's even more likely that they made COVID"?
I mean, you can ask that with any question. Like, imagine this, imagine that. I don't know. I'd have to examine like what exactly the strain was, and I mean, obviously, this is not a key point on which the lab leak hinges or the zoonosis hinges. It's just to date, it's what this is what the alignment hinges on, because and all of the aligners, first of all, we don't have any sequences that have the deletion without the insertion, the claimed insertion, and all of the aligners just more think it's more parsimonious to align this as nucleotide mutations. And
Yeah.
Okay, let me.
We're kind of beating this to death. Maybe we should move on to other points. Unless you have something else.
I want to ask, so the one other question I want to ask, which contrasts both of your theories about the plausibility of naturally finding this versus injury or whatever, why not a competition experiment? So this seems like the perfect kind of thing to me for you stick a bunch of pieces together, or you do a whole bunch of competitions of your viral constructs in a dish, and then the one that pops out is, for evolutionary reasons or conserved reasons, the one that's good. It seems like that would answer in some capacity, Peter, your question about combinatorial unreasonability. Can you speculate on that?
I mean, they have to make, if you're talking backbones, they have to make the reverse genetic system for that backbone to begin with. And that takes a certain amount of work. I think when Yuri and Stuart debated, they said it was like 3 months, but then groups since then have done it faster. And like,
2 weeks.
what's the fastest they've done, Yuri? Like a month? Less than
3
a month?
weeks. Yeah, initially like
3 weeks?
3 weeks is the fastest.
3 weeks. Okay, so even if they're like their groups are really good and they want to try all of these, with a different, so they've got first they got to make the 180, reverse genetics systems, and then they have to try all these combinations. And I don't know if you can like put them all in a petri dish and compete them all at the same time. I'm not sure how that works. Plus, it's going to have the cell culture adaptations because we don't even know which cell culture really works for this. So, I mean, possible. I don't think that's probable.
And then, Yuri, will you speculate, do you think that competition experiments, or I'm thinking of some sort of combinatorial Golden Gate assembly here. I think there are ways that you could potentially do these things that don't mean, you're taking advantage of the combinations being produced by and amplified by evolution. What do you think that that's part of it, or do you think that they actually mixed and matched by hand?
Like if we're talking under a lab leak scenario, whether they mixed and matched by hand or had some sort of competition experiment where they just, put some building blocks in the cell culture and kind of let evolution figure out the best scenarios and see, which virus would then emerge to like get amplified in passage through cell culture experiments. It's an interesting hypothesis that could have been one of the ways that this could have been produced.
I'm just curious why you guys have not brought that up, just to in bacteriology, like competition experiments with TNC mutant libraries, or those are like very, very common. So I was just wondering if there was a reason why that was not part of this.
I just haven't heard of this being used in virology, to be honest, for like this type of.
Totally. Okay.
But it's an interesting hypothesis.
I guess for me, this is also just relative probabilities. Like, Yuri's got a bunch of reasons why the proline could be there. Maybe it's MERS, maybe it's they saw one of these viruses and they recognize it somehow. Maybe it's because there's a feline coronavirus, and like maybe Xi Zhengli really likes cats and she built a virus with that. But I don't think that's likely. And I think when you come up with that many different explanations, that doesn't necessarily help your theory. Also. Maybe moving on, just a few things I wrote down while Yuri was going through. Like the picture here, another thing to note about this picture is January 15th, 2020. So this is after like the virus is already leaked it's loose in Wuhan, and they know that, because the market outbreak's been announced. It's like a week before the Wuhan quarantine, and they've all gone out to dinner, no masks, no concern. So SAR would call this human inference, but this is one of those points of like, who actually knows? Like, this is Ben Hu right there. Like, so he just recovered from COVID and he went to the hospital, and one version of David Asher's story is his wife died, and like he's, smiling with his colleagues. Do you think maybe they should be acting differently if they created the pandemic, Yuri?
Or maybe not. Maybe they're supposed to carry on business as usual, right? So maybe I think you can bring both sides of the argument to say they had to, not show any signs that there's anything out of the ordinary happened in the lab.
Yeah.
Yeah, I can invent any number of plausible explanations for, a picture.
Sure, but we've been looking at this lab for 4 years now, right? So, and we have intelligence agencies and really good sleuths and drastic who've found a lot of interesting things. But should they maybe have found something by now? And the intelligence agencies can hack people's email. They can, if there were all these secret sampling trips, maybe there'd be evidence that they went to Laos. That they obviously we don't know.
They went to Laos.
They went to Laos in 2014, right? Did they ever go back to Laos?
No, I think 2016 is the EcoHealth. Like the EcoHealth published a report showing that they had Laos samples, and of course, 2017 paper by the Wuhanist virology mentioning that they collected viruses on the border with Lao, TPR.
So if it's like I think these viruses were from 2014, then you have to imagine that they like sat on them for 6 years and then didn't publish them at all, or you need to imagine that they had like secret chips in 2019 that.
Well, it didn't have to be in secret. They could have had samples they didn't even sequence, like some stuff they collected in 2014 they didn't work on until 2018 or 2019, and they didn't analyze the sequences. So it's not like any secret. It's just like you have a lot more to do stuff during research than you ever get to. So,
but like they wrote a bunch of theses about, or they mentioned RATG13 in various experiments, like the 2018 one that came out, and they had no reason to hide that. So why would they hide the secret one that's closer than RATG13?
What do you mean hide? If they
But I mean,
if they
didn't
caused the
really-
pandemic, obviously they would
They didn't
hide.
release it in Latin, so yeah, so either they collected it like really recently compared to the 2019 paper, that's Latin, or they have no reason to hide it back in before the pandemic. So
Yeah.
do you think there's just a lag
No,
between?
they would have no reason to publish it. If they didn't work on it, why would they publish it?
Why did they say 180 viruses in diffuse? And why is that the right number that matches their other?
No, that was the
Like-
number in 2018.
Yeah, and that matches the papers at the time, and
That
then the number for
matches
2019
the
is
EcoHealth.
slightly higher. So how many new viruses do you think they collected between that 180 and the pandemic?
I mean, that paper was by EcoHealth, right? So it's in the diffuse. EcoHealth was a co-author, so I think those are the viruses that they shared between EcoHealth and WIV, and maybe some of them were collected by EcoHealth originally and sent to Wuhan for processing.
So how, I mean,
think
diffuse,
this kind of
diffuse says,
argument that why would they not publish everything? I think
yeah, but we can bound it, right? Because diffuse says they're going to collect 3,000 samples, and we know that's going to be like 30 sarbecoviruses. And so if they had 180 and
if you
the
said
normal ones,
like 10,000 samples between 6 South Asian countries, they plan to collect. But I mean,
page 31 said 3,000 samples. Maybe I have the wrong page in there. And we know the rate of you find sarbecoviruses, so that's like 30 of them. So maybe they have 180, and if they did diffuse, they have another 30. So then, like, we have to figure out the odds. What are the odds that one of those is like Banal 52, right? So that's like, if you go to exactly the right part of Laos, it's about 1 in 100, you'll find one of those. But if you go to the wrong place, in the grant, it says they're going to go back to their cave in Yunnan, and they've sampled that cave like 100, 200 times already, and they never found one that would work to make COVID. So if you find 30 more in that cave, you're not going to find another one. So you need them to go to like a different cave, and you can work out odds from that. You can put some, if you're doing a Bayesian analysis, you need to put some number on the odds they have a secret virus. You can't just assume it's 100%.
well, you're just kind of basing everything they would ever do on diffuse. And they had many more collaborations, including with the Guangdong Institute of Applied Biological Research, who have collected more viruses. They did more sampling, including for WIV, and they had basically, that's where the pangolin MP789 comes from, and potentially the some sampling, and of course they sent samples to the pangolin samples to Wuhan to work on them, so they shared samples like between the two, and they wouldn't have published them until they had any results to publish them, and we only see work start to come out now on these pangolin samples, like.
I mean, that might be possible, but then it's an even bigger plot to cover up. So, like, I believe in small conspiracies. If it's just like Ben Hu and Xi Zhengli and nobody else knows, they can hide a secret. But now you're talking like multiple labs that are sharing their viruses, and yet no whistleblower and no ability to figure that out. That's getting more.
Can I pause this and to try to? I'm sorry, I'm getting lost on the counterclaims. I don't know if Eric, this makes complete sense to you, but I would like to.
It
I
does
would like
not.
to again see if I can summarize what each of you are saying. So, I'm hearing Yuri say that there are likely a large number of samples that were never sequenced. They took a bunch of sampling trips. They're recorded in somebody's notebook. They get sequenced and used as research involving them comes up. It would not be implausible to think that all of the releases of information in various different papers at various points did not include a large number of these. Whether or not diffuse bounds the number that are being additionally sequenced, that doesn't really tell us anything about how much WIV had on its own. Yuri, does that accurately capture what you're saying?
Yeah, pretty much.
Okay. And Peter, I'm hearing you say that all of the existing disclosures, including the 2018 GenBank submissions, as well as diffuse, generally point to there not being a whole bunch of these other viruses around. Even the sampling numbers that they give for like raw bat poop samples would still put the number at maybe being able to recover 30 live viruses or something like that. So the probability that they had a large number of secret backbones is very low. Is that a fair?
Yeah, and I'm trying to predict. Well, first off, I'm trying to predict who to listen to, who to believe. And so if Xi Zhengli is saying, I don't have more than 200 viruses, and then we get these FOIA grants and uncovered papers that confirm that number, maybe I should believe Xizheng Li because she was correct. And if Drastic is saying, RATG is secretly 4991, it's a different virus, and they're wrong, and then say there's these other 8 secret viruses from the same Mojang mine, and they're wrong, and now Drastic is saying, well, they're secret viruses from Diffuse. I mean, one of those theories has had predictive validity so far, and one hasn't. So I'm probably going to trust Xizheng Li more than I'm going to trust Drastic at this point.
It's very different.
There's no difference. There's no proof of secret viruses yet. But I'm trying to take this a step further because I like to, steel man the other side. And I'm saying, okay, let's assume that Diffuse happened, and let's use the numbers in Diffuse and how many secret viruses could they have? Because, Rootclaim wants to have this probabilistic argument, so I have to turn everything into probabilities. So I said, okay, it looks like they can maybe find another 30, and what are the odds one's going to be close enough? And I went a little further with 15% odds you'll find one that's like RATG 13 in those 30, but RATG 13 isn't good enough. You need the RBD. And if it really needs to be like Banal 52, you pick 30, there's 1,500 coronaviruses. It's only like 2% you're going to find a Banal 52. And that's still only like 97%. Maybe you have to adjust that downwards. And then I adjusted it downwards like another, factor of 10 just for the no secret thing has uncovered it. But you could take some steps out of that. I'm just trying to put can I give a number on this? Can I estimate the odds they have this? And that's what I'm using in my analysis.
Okay, Yuri, you took exception to a part of that. What was that? Or no?
Well, I think just kind of coming up with things to reduce the odds by. I think it can go on forever. And if they had 30 new viruses where the odds one of those viruses could have been the progenitor of SARS-2, I don't think you need, what you keep reducing is from, I guess, hindsight that you'd need this and you'd need that, but it's
But we know for sure they need those things. We know they need the RBD, and we know they need...
But it's not like they would be looking, going out in retrospect, just if they were sampling viruses in the locations that we now know house these Banal-like viruses. And also we know that, for example, RM102 came from a cave in 2019 where Giabra was sampling 15 kilometers away, looking at these Rhinolophus malayanus bats, which are the ones that have Banal 52 in them. So the odds of, say, Giabra, that person, Li Biaozhang, who collaborated with the Wuhan Institute of Virology, finding some sort of progenitor, or maybe just even getting samples and sending those samples to the WIV, and WIV then extracting some genomic sequences that have similar viruses to Banal 52. I think, those odds are in the kind of realm of possibility. You can...
But we don't know where they sampled. So you're assuming first off that they went to the right part of Laos, not that they went to their cave, which doesn't have to be viruses.
It can well be in Yunnan, because RM102 is from Yunnan. It's from but
not the RBD. There's BTSY02. I don't know how you can.
BTSY2 is from Yunnan and sampled in 2018.
Yeah, so there's 5 of these that are have the RBD, and only one's close enough to be COVID, which is Banal 52. I don't understand how this Bayesian analysis doesn't think about these questions. Why do you just assume 100% that they had the virus? That's not Bayesian reasoning. That's ignoring a key part of the question.
Well, Bayesian reasoning is SARS thing, so Rootclaim's thing.
Okay.
So
yeah,
I'm not.
Well, so how do you look at it? Why do you think it's more likely? Or sorry, one other question I'm going to bring up here. You're talking about GIABR, and they're in what, Guangzhou, Guangdong? Yeah. So if you think they have the samples, that means you could make a lab leak theory if it showed up in Guangdong also. So you've lost the part where Wuhan is unique. And you've done this a few times on Twitter where you're like, oh, well, they have the pangolin viruses in Guangdong, so maybe that lab's involved.
Well, if only that happened, your lab leak would have been this kind of argument. Yeah, you can go on forever. on the other hand, if you had any evidence of zoonosis, I'd like to see it. That would close all the questions. So, if but actually, before we got into this discussion, you made a point I wanted to respond to about the FCS. They would not have picked this FCS. They would have just inserted RRKR, which is a really good cleavage site. And actually, I think it's very likely that, again, they would have had a panel of cleavage sites, because I think it's not just so straightforward that like the best cleavage makes the best virus. Because you actually talked about this in your presentation, that furin can actually pre-cleave these spikes in the endoplasmic reticulum, if I remember correctly, or the Golgi apparatus. I'm not sure, but basically within the cell before it gets released. And if you have a really good cleavage, you can end up like with, because it's a trimer, right? It's a 3 copies of the spike protein on the virion, and if you cleave all 3, you lost the thing that binds to ACE2, and it's only good for like creating these Synthia cell like nearby cells. But if you actually have like moderate cleavage, you can maybe cleave one of them, and this kind of opens up, and you can infect both nearby cells, but still the thing that binds to ACE2 remains on the virion. So maybe actually like the most optimal cleavage from a viral fitness standpoint is not the most highest cleavability score for prop1. There could be like this Goldilocks zone where it's like, still cleavable, but not too cleavable, and that can actually change viral tropism. Like if it's cleaved and host tropism, like if all of them are chopped off before the virus leaves the cell, there goes the end of your transmissibility for the virus. So
but it is observed that COVID has evolved different cleavage sites as time has gone on?
No, only before the proline, the only one that mutated, but that doesn't. I don't think, there's actually disagreement where proline increases cleavability or actually decreases cleavability. And if it does, it's again with your prop 1 scores, it's actually very mild difference.
It's not a disagreement though, because we've seen this in nature. It turned from P into H with alpha, and then it turned into R with delta, and every time the prop score got better and the variant got better. So you can hypothesize that the cleavage site could be too good, and then that's going to be detrimental. But has there been an experiment showing that? Like so far, it's only gotten better, and it's only been better for the virus.
Well, it hasn't gotten better. Like the cleavage site remains. Like it's RRAR rather than it didn't evolve into RRKR or RRRR.
The position in front matters, and it influences how good of a cleavage site it is, and we've seen that.
The leading proline or whatever it is in delta. It influences a little bit, but I wasn't talking about the proline. I was talking about why didn't they pick the best cleavage site for cleavage?
But we don't. Have you seen an experiment saying that if it's too good of a cleavage site, it's detrimental? You're hypothesizing that.
Yeah, this is just a hypothesis I have.
I'm sorry. I thought Peter, you showed evidence of the cleavage. If like the ribosomal pausing. I'm sorry, did I misunderstand? I thought you said if the cleavage site was too good.
That's the CGG, right?
That's the CGG. Yeah. Right now we're just talking about the amino acids, and there is no. So far, we haven't found one that's too good. It's only gotten better, and in each one, alpha took off, and delta was, took off, and it killed more people, and Omicron is a little hard to compare because a lot of things changed with Omicron, but it's still an even better cleavage site.
How much of this in this infectivity can you confidently assign to those changes? There were presumably other changes between those things.
In Omicron, it's hard because it's like the immune evasion on that is the big factor, and not this stuff. Maybe it just happens to have a better. These other ones, I think this is a big part of all of these, like. The 614G, that's entirely the cleavage site. That's why that took off. And these other ones, were there other issues with alpha and delta? I'm not sure. I think the cleavage site was a big part of each of
alpha and delta have the same cleavability score, despite the mutation.
Yeah, you're right. So the score's not 100% predictive, but we're not like finding what you said about like the cleavage can be too good. Like that's possible, but that's it's a hypothesis.
Yeah. That was just a thought for why they would evaluate several different potential cleavage sites.
And if they also evaluated several different ones, why did the bad one leak? Like if they made all of these, if they just had some apparatus that could make like 100 different cleavage sites, why wouldn't the best one leak?
It's not I mean, it's still the same cleavage site. We're just talking about the proline, which precedes the cleavage site. This cleavage
Okay, well, the
site
alanine, then. If they tried 20 different things for the alanine, why didn't the one with...
it might actually be the most optimal. Like this is the Goldilocks.
It might be, sure. I mean, get a lab and try that and publish that. It'd be a great experiment.
Yeah. Well, let's do
I mean,
some gain of
that would
function.
help.
Let's see
That would help establish
what
the...
else we can cook up. Yeah.
Do you want
Yeah,
to?
and don't leak that.
Our,
Do you think Omicron was a lab leak, by the way, though, Yuri? Like,
yeah, definitely initially it was suspicious. I didn't. I think what reduced my thinking it could be a lab leak is all these cryptic wastewater strains that people are finding in like Ohio, which have like because
they're still
basically
Delta.
sorry.
Like the ones that are still Delta cryptic strains.
Yeah. So like before Omicron, we just didn't have any plausible way for a virus to acquire so many mutations. Like we couldn't even think of like even in immunocompromised people, they still didn't acquire as many mutations as Omicron. Just kind of jumped out and had 33 extra mutations in the spike. So and of course it's exactly like the synthetic polymutants that they had in a lab, where they just took all the best hits that they generated through subjecting SARS-2 to immune system pressure they used like antibodies from various.
Yeah.
But anyways,
those experiments are kind of terrifying. Like those are gain of function. If you're trying to find something that evades the immune system.
exactly.
But nature does the same thing. Like we have 100 million AIDS patients in Southern Africa. And I don't think it's a surprise that Omicron came from South Africa, because you have all these people with immunocompromisation, and then they get, latent COVID infections, and one of them has an immune escape variant.
Yeah, except
That's how I would
in all
explain
of the known cases of latent HIV infections, like co-infections, HIV, or other immunocompromised people infected with SARS-2 for like 18 months, they still didn't get as many infections. Like the latent virus didn't get as many infections.
But it lasted for like a year, and it got a bunch of mutations.
But it had to have had because the original strain from which it diverged was like a November or something. So basically, it only had like 12 months versus.
Yeah.
But I think we're getting a little side.
Yeah, let's kill this topic. One I wanted to talk about is again, would they recognize this as important based on what they wrote in Diffuse? So you highlighted a bunch of things. I read through Diffuse again this weekend, and I was thinking, you can highlight other things. Like they point out these are BSL-3 viruses, so maybe they would work with them in BSL-3. But they also, even more importantly, they wrote that they were talking about recovery of full-length SARS-CoVs, and they want SARS-CoVs that are less than 5% nucleotide variation. I don't know if I can...
it's 5% nucleotide variation within
5% from what? I think that means 5% from SARS or from like WIV1, because they're interested in these. Because WIV1 is 96% from SARS, and so that's what I'm saying about COVID is 80%, and why would they care about COVID? Because it's so far from SARS. It's not one of the full-length ones they're going to recover.
No. I think you're misreading this, because this is saying we'll compile sequence data from closely related strains, and they're then explaining what closely related strains are. They're the strains with less than 5% nucleotide variations, and then they'll compare full-length genomes, scanning for unique single nucleotide polymorphisms representing sequencing errors. Basically, they have this quasi-species, whatever they're defining, like closely related viruses in this kind of cloud circulating in a given bat habitat, that they will then sequence and see which ones are the more prevalent and which ones are the less prevalent, and they will then look, are the less prevalent really actual less prevalent viruses, or maybe it's just sequencing errors. And then when they talk about these like high risk and low risk strains, basically they're saying there's like some low number of strains, a low number of variations within the same kind of quasi-species that might have dangerous mutations. And I think this actually comes from like an alpha coronavirus paper from like 2018 that looked at the bovine coronavirus, which they sequenced like to a very, very high depth, and they noticed that there are sometimes they have actual cleavage sites in these bovine coronaviruses emerge.
Yeah, that was in the Slack messages, right? That they were talking about.
Was it
passaging, and then they found the 4 nucleotide or the 4 amino acid insert in the bovine coronaviruses.
Maybe that's what they were referencing.
2013 experiment, is that?
I think so. Yeah. So, but that was an
And
alpha.
so it was already there in the bovine coronavirus, but then when you passaged it, it got increased in frequency. If we're
Yeah,
talking about the same.
exactly. And also, this is, I think, is similar to what they're saying sometimes happens with feline coronaviruses. That you have basically happens what turns a regular feline coronavirus into this infectious peritonitis. Basically, it starts mutating and mutates a novel furin cleavage site, which makes it lethal.
So do you think these things could exist in sarbecoviruses at low frequency, and then it gets into the right species, raccoon dog or whatever, and then that gets amplified to become a selective
it's
advantage?
definitely theoretically possible, and I think this is what they were looking for. And this is the passage that you're quoting is basically showing that they're trying to find this evidence of this in sarbecoviruses. I don't think to date anybody has found it.
Yeah.
And
So,
obviously, like to me, coming back to the most plausible natural way this furin cleavage site would have arisen through this insertion is exactly with some sort of recombining with the host mRNA, and then creating this, like initially a very low prevalence kind of subspecies within this cloud. And in bats, the problem is in bats, this would be selected against.
It's selected against.
Yeah.
We're agreed on that. Yeah, it's going to happen in the intermediate species, it's more likely to happen, much more likely.
Yeah, so the chances of then that kind of first, it's a bit of a chicken and egg. So for this furin cleavage site to emerge in this way, it would first need to get into this intermediate host like a pangolin, but without an FCS, would it even infect a pangolin? And then the pangolin would need to evolve this.
It can be fecal oral transmission on a farm until it becomes respiratory transmission, or
Yeah, definitely. Initially could be, but yeah, you need like a pangolin in close proximity with bats, with these infected by these viruses, and I mean, yeah, like this. That's why I've looked for host mRNA fragments, sequences that could potentially serve as the origin of this insertion, and found it in this pangolin sequencing data set. So to me, this would be the only, well, not the only, currently the most plausible way this could arise naturally. But of course, again, we don't really see any evidence of this. It's just like a hypothetical idea.
What I'm reading here is the 5% means difference from SARS, because everything else in this talks about WIV1 and WIV16, and those ones are 4% from SARS.
Not in this
and they have a picture here that says spike similarity. They're going to look for that, and I'm feeling like that's spike similarity relative to SARS and ACE2 interaction. So I'm not sure they're going to spot SARS-2 precursor as important, because the spike's not similar. Maybe they can spot it here. Maybe if they have a good enough model, they can see the RBD is good.
Just above, it says that SHCO14 has 88% spike similarity to SARS-2, SARS-1. So it's still interesting for them. So
and the grant said WIV1 and 16, I think. So
So
And
I mean,
those are also the backbones they have. So they're going to use the backbones they have because that's easier than making new backbones.
No, I don't think 5% referred to similarity to SARS. I think 5% referred to like this.
I would move to pause this particular thing. Eric,
That's
do you?
problem with these debates. You
No.
start like going into not a problem, but that's you start
No,
going
this
into
is
something very
fun, like-
deep, and it just takes hours and
You know,
hours to.
you know the answer. You have answers. And like last week, it was like SAR hasn't researched this very much, so it's really frustrating to. Like most conspiracy theorists are like, I did my own research, and like SAR is like, I didn't do any research. I just multiplied numbers together, and so he's hard to talk to. But this is fun. This like I first heard Lab Leak on a podcast with you on it, and so this is like I finally got to the boss level.
I had a question. So you said you were initially for the lab leak. So what turned you against the lab leak?
Oh, I thought Drastic was going to solve it, or the government, or whatever. It seems so complicated. I mean, the judges are probably here just like neck deep in this stuff trying to figure it out. And I was like, I don't want to go through this. I'll just wait it out. And, I finally said it's not going anywhere. It's very polarized. Let me look into it myself. And, I read the Warby paper. I read the PRRAR paper. I looked at, these cleavage sites, and I learned what a furin cleavage site is. That was certainly like originally they're saying it's the unicorn's horn, and I'm like, oh, it's a perfect unicorn's horn, but it's actually not that at all. It's like the previous experiments, put this gleaming white horn on the unicorn, and this one's like it's curved and it's green and it's out of frame, so they like put it over one eye, not like right in the center. It's like it still gets the job done, but it's not. Evolution does weird things like that.
The unicorn.
In a long process, and after about a month, I was really leaning against Lab Leak, and it's just gotten stronger as I've dug more and more into that. Like, I feel like Picard's research holds up pretty well, and I feel like maybe we should transition over to that next, and talk about like your different theories on Lineage A.
very convenient. I have a question.
Okay, maybe I should
But-
yield the screen, or
It's fine.
or is it for me or Yuri?
It's going to be for you, Peter.
Okay.
And it might not be exactly, but it has relates to Picard stuff. I'll just quickly say I can only read this less than 5% nucleotide variation the way Yuri interpreted it, as they're talking about variation within a panel. I
Okay.
realize that sentence could be ambiguous. That's just how it looks to me, and I think interpreting I mean variation from SARS-1. I mean, that could be what they meant. I don't see it. That's not how I would read the sentence naturally.
Let me just chime in.
I might be confused there. I mean, so when they say 3 to 5 full-length viruses per year, what do you think they're going to prioritize?
Let me just chime in before that question is answered. Just like the reason I don't find this debate particularly important to resolve is because no matter what we all say here, I don't know how many grants you all have written, but the relationship between proposed grant work and carried out grant work is tenuous at best. So like it's very hard for me to understand why that particular debate, like how the
That's a very good
5%
point.
matters. Like no matter who wins that debate, I do not think it's going to be dispositive for me. Not to say that it won't be for you, Eric, but I just don't.
No,
I talked to one scientist about this, and they said this is probably like some 22-year-old working in Barrick's lab that's hoping to go to med school someday, and they've listened to a few talks on protease cleavage sites and don't actually really know what they're writing here. So it's hard to...
I don't think that that's true. I mean, just because, this is a gigantic grant, and like when you're cutting these things down, right, you're looking for every word. Like, I think it's much more likely that this grant is misleading because they had to cut it down to size. Like, if you've ever written an R01 or an SBIR grant or whatever, you're bumping up against the limits and you're fighting people who have spent, thousands of hours to craft their grant. So I'm just like, I just don't know how much grant viewing is going to tell us about what actually happened. And it also seems consistent with Yuri's position where it's like, well, this diffuse might be a part of the like intellectual milieu, but it isn't necessarily what happened. Like, I don't know why we're focused on this.
Right.
I don't want it, like, Eric, obviously it's important. So I'm not trying to like belittle that. It's just,
No, that's a very good point. I mean, Peter's right. In general, would prefer to look at viruses similar to SARS-1, but I don't think we can definitively say, this virus is too far.
We're already relying on a grant that says they're going to do work in North Carolina to assume what's happening in Wuhan, assuming that they have the technology that's in North Carolina, And we're going on from that to say, well, it's not really, this grant really couldn't make SARS-2, but maybe if they went beyond that, I mean, that's speculation on top of speculation.
I definitely
What technology?
want.
Like inserting a furin cleavage site doesn't require.
No, but like the
technology.
HAE cells, you go through and they've never used those before. So that's another step in there. That,
Well, I mean, using primary cultures, I think they actually used. Well, in any case, yeah, like, but using a cell culture is not like a technology. And they definitely had humanized mice. Barrick sent the humanized mice to Wuhan in like 20
yeah,
whatever.
but those don't work because of the 501Y mutation. That wouldn't make COVID. So you're really restricted by. That's why I looked into all these culturing experiments. Like, how could they make it? And also, like, if it did make, if you did use the mice, it could leak. It would just be a different virus because it would have the 501Y mutation.
Like, yeah,
So
this
they did
claim
that first,
that
and
it
it leaked
necessarily would have absolutely had the 501 if it leaked from mice.
It's like the second generation mutation. Like it happens really fast in the serial passaging. So, it could leak in the first generation. And maybe that's even a more likely way for it to leak because like we have these leaks from Taiwan, right, where it actually got out of SARS-2 got out of a lab, but they were like working with mice each time.
Mouse bite.
And
Maybe,
so
I was going to ask a different question, but let's go to this then. Okay, so you talked several times about how, you don't think that SARS-2 could have been cultured in a specific way because when people did that actual experiment, they observed specific mutations which are not present in SARS-CoV-2. And to me, it's not obvious that, this is consistent and replicable. But now you're saying if you attempt to passage SARS-CoV-2 in humanized mice, that consistently within 2 generations, a specific mutation should have.
I can pull up the graph. But yeah, it's in like the second to third. They measure how it fixes the mutation over time,
Yeah,
and it
can
gets
you
fixed
just very quickly show that to us?
Yeah,
Because,
and that's true of a lot of these things. Like in the Vero cells, when you passage it in those, it loses the furin cleavage site really fast.
And by really fast, you're saying 2 generations, and by generation we're talking about passage from one
Passages.
animal to another
Yeah.
is a generation. All right, let's see this graph, and
Thanks.
one of these days I'll ask my other question.
Sorry, that was my fault there.
No,
Okay, so sorry, it's not as fast as I'm saying. Second generation, zeroth passage, first passage, it's at 50% on the second passage, and by the third passage, you're at 90% fixed with this
but
mutation.
am I reading that correctly in saying that the virus that they initially introduced already had some proportion had this T mutation?
Yeah, maybe that. I don't know if that means that there's always mutations at the site and it's sort of
Is that
selectively
even
pressured.
SARS-2?
This is SARS-2 in humanized mice, yeah.
Like my guess, without reading anything, just looking at the picture, I read that to mean after 0 passages, so before introducing, 15% was T. That's what that looks like to me.
Well, so that might be you put the virus in the mouse and then you haven't passaged it. And so it's already mutating in some of those mice and just introducing it. I believe that would. And so it's like 10% just from the
I guess
introduction.
the reason I'm making such detail over such a distinction is because there's a very big difference between this mutation arises de novo consistently within 2 generations and this mutation, if it's already present, takes over.
Well, so it's not already present because these mice don't have COVID already. And presumably, if it's a lab, they're using a pure strain from whatever sequence they've.
I mean, I would use a pure strain, but the P0 column is not pure, so.
So P0, they've infected these mice and then they've measured it. So some of them are going to have a mutation before you even passage it.
Okay, I'd have to look at the paper to.
And so this doesn't prove it's not a lab leak because you can still get the mouse bite in the first or second generation. But
Sure,
if you've done a lot of work with this, you're going to find this. And if you do a whole lot of work at the lab, probabilistically one of these is going to leak. And what are the odds it's the first versus the second? That's how you'd look at it.
Does Yuri want to comment on this, or
let me try and just sharpen this. I think the thing I don't understand is this is one mouse experiment in one mouse context, one set of isolators or something like that. Is there is this something that is reproducible? Like, I understand there's lots of experiments going on of other fixative mutations. How has this been reproduced?
I do not know. I have not consulted the entire literature on humanized mice experiments. I know that you can't do that if they're not the humanized mice. COVID doesn't work at all. They're not. You can't infect them with it. So you can rule out the normal lab mice, but obviously they use the humanized ones.
It's just like one experiment has seen this particular mutation become fixed in the population. It's
well, so like the Vero cells thing is reproducible. Like Vero cells.
I'm not on the other things. Just this.
I haven't reviewed I looked at like 8 different mink evolution experiments to try to understand that, and I've looked at, it's a huge subject, so I haven't gone into huge depth in every field. So,
Yuri, do you want to comment on humanized mice before I finally ask my question?
No, I think it's a good point that it'd be interesting to have a panel of experiments to see how often this mutation actually arises, because it seemed to
So after you build your gain of function lab, you should try that after you try the better cleavage site.
Taking down the list of the gain of function experiments to try. So go ahead, Eric. You had a question.
SARS-CoV-3 overall. Okay, so Peter, I wanted to ask about the question of 2 spillovers. So we're talking about this this unusual structure for a phylogenate tree to have 2 branches, and I think you were quoting, I forget the name, Descartes, had modeled and argued that it's unlikely to have a single root that has 2 significant subtrees that are not descended from each other. And what I don't understand is how having separate spillovers makes this more probable, because you're saying, if you're speculating possibly that lineage A and lineage B, neither is descended from the other, and they are separate spillover events, but that still results in this phylogeny. It's just that the most recent common ancestor is now an animal instead of a human. In my mind, so whichever one.
Yeah, I get exactly what you're asking, and why can't it be 2 mutations in an animal but not in a human? And I think the main difference there is if it's 2 mutations in a human, that usually happens 1 and then 1, and you see evidence of the intermediates over time. And the rates are something like 60% of people infect somebody else with COVID with 0 mutations, and like 30% infect with 1 mutation, and like 10% infect with 2 mutations, something like that. I'm not sure if that's exactly the rate they used in Picard's paper. So it's more likely that you're going to get like 2 as 1 and 1. And generally what happens when you look in the real world is you always find those intermediate, or you frequently find those intermediate jumps. And why is it more likely to happen with the animals? I mean, part of it's that at the beginning, you probably have a larger number of infected animals than people at the very, very beginning. And so you have more opportunities there. And the other thing you might be missing here is it's not just the phylogeny, it's the AB, it's the genetic clock. It's the fact that B looks older than A. And so that's really hard to explain with the human model, where A spills over first, but then it just like sits and does nothing for a week and then becomes B and then explodes, and then A starts growing. That's very unlikely. Whereas with the animals, you can have B spills over first and then A, and that can explain both the weird phylogeny but also the molecular clock, because the intermediates are hidden and the timing can break that way because the timing, yeah.
I mean, I still don't see how model B here becomes more likely by making the blue diamond an animal instead of a human.
Oh, so B's not likely either way. C is what we're talking about. We're talking about normally it's A, and C is like this is B, and that's A.
If lineage A and lineage B are separate spillover events, then aren't we getting picture B here?
So this is assuming it's got 1 mutation from each.
I guess what I'm saying, the only way to avoid picture B is if either lineage A is descended from B or vice versa, right? If they are not descended from each other, then we're looking at the middle picture. That's my understanding. I could be misunderstanding.
Yeah, I mean, I'm trying to explain this better. There's these 2 mutations, both are like a C and a T that changed. And here he's assuming the the human intermediate, assume the human intermediate had a C and a C in both spots, and in one of them it turns into a T, and in the other one it turns into the other T, and then those both spread equally. And he's saying that basically never happens in his simulations. And this other one is his simulation saying, here you start with lineage B, and one of its descendants has two mutations at once, and it grows into this big clade. And that does happen sometimes.
Right. I mean, I don't know if Will or Yuri want to chime in. Because I'm not talking about what specific mutations, I'm just asking the simple question of who's an ancestor of whom.
I had this image in week 1 that might make it clearer why it works with animals.
All right, let's do this. Because I remember when we talked about this week 1, I wasn't convinced either. About the two spillovers explaining things very well.
I mean, yeah, even if they were two spillovers, they had to happen at the market. If we're talking zoonosis.
Yeah, they happen.
And we have zero. Well, one sample in the market that I actually wanted to talk about, but
we do actually have 2 Lineage A samples at the market. If you look at the small levels of reads, that's an interesting one we can get into.
Do we want to?
Right, you said 25 in your presentation. Where was the 25 number coming from?
25.
You said there were 25 environmental samples that were sequenced.
Yeah, basically the Gao paper and the PRRAR preprint. I'm looking at right now at the actual table. Okay, they have like.
All right, should we talk about A and B at the
So,
market?
like this.
Yeah, exactly. Yeah, that's what I'm looking
So
at.
we all, like, everyone disagrees. Rootclaim said there were 923 samples at the market, but that's obviously they're not all sequenced, is why he was confused.
Yeah.
And SARS
they were
only,
collected, and out of those, only like 70 were
so
positive.
a bunch of them got sequenced differently, and only a few of them had like full coverage or close to full coverage. And I went through to see but some of these are like half coverage. So I went through to see if any of those half coverage ones you could tell, like that could be an intermediate, but you can still guess whether it's A or B from that. And I made a full table of those. And so you have one of those half coverage ones has Lineage A positions. It's only like 2 samples, but so it's probably 2 out of 11 samples are Lineage A. It's not just the A20 sample.
Well, it's just 2 reads, and it's
it's 2 reads, but the other ones are like 3 reads or whatever, and there's no coverage at the other. So you can't. That could be an intermediate, or it could be random. So that's not like strong evidence for it. But there's only 5 that actually got sequenced that you can tell because they have coverage on both. It's not 25. So it's 1 out of 5 that are Lineage A.
Sorry, what? 1 of 5? Oh, you mean 5 that have coverage of the region? Okay.
Both sites. Yeah, you said 25. It's 5.
No,
so
no,
you
I
can't.
said 25 in total that had, but yeah, obviously it's more than 25. I think it's more like 30 that actually had, that were sequenced and had any reads for SARS-2. But out of those,
Most of them had no coverage at
most
those
of them
sites
had
at
no
all.
coverage. Yeah.
Yeah.
And
So this is what we have like 11 with coverage, and you can of those it's like 1 or 2 is Lineage A.
When you say coverage at the sites, you're talking about the sites where A and B differ.
Yeah, A782 and 281. Those are the 2 sites that differ.
And like if you actually look at that first table, you see that like A20, like it says sampling type SARS-2 amplicon, and it has like 10 million reads versus the next closest from cell culture has 2 million reads. And I think it actually in the Christoph paper preprint, they mention it's also from a cell culture, like they try to culture it from the glove. So, and of course the A20.
I think in Lou it says it's actually not cultured. They just resequenced it with amplicon.
It's so weird that the next kind of like if they resequence so it has 10 million reads, and the next closest one from
It's 2 million.
But the 2 million is from actual cultured sample.
Yeah, or Lou got it wrong and said it was cultured, and maybe he got it
I'm pretty sure
wrong.
it was cultured. I think I saw it. I didn't reread the paper. I didn't have time now, but I'm pretty sure that Christoph.
One thing I want to point out here for Will, because he was asking at the first debate about like how come there's only 1 to 8 samples in the raccoon dog samples and how is that real? A20, this sample, it was 22 reads when they just sampled it the normal way, but then when they resampled it with the amplicon sequencing, it's like 10 million. So there is a possibility that they could, if they still have these. You were also asking like, do they have more data? Could we solve this with? Like if any of these samples are still on ice or something, maybe they could resequence them and we get a little more data that way.
I'm sorry, the SARS-2 amplicon that's just not like the ribosomal region. That's the full. They're amplifying the full genome with.
I believe so. I'm not a sequencing expert, but I think it's like a primer.
If they're covering all the bases, that must be okay. It's just that's interesting.
Yeah, and so that actually like if that sample can vary by a factor 22 versus 10 million. Then the actual number, like, oh, it's only 8 reads or whatever on the cage sample, I think maybe that's not as good of an objection. It might just be the sequencing method sucks.
Okay, there's something here that I just don't know. What does read count mean?
Method, they had like 200 million reads on those cages. They actually used, I think.
There's actually a reason that I looked this up, because it's something more interesting. Like, everybody, like every time the lab leak finds something they don't like, they come up with like as many objections as they can for why they don't like it. And so there's like the A sample at the market, and they don't want A to be at the market because it can still be a lab leak if Lineage A started at the lab. So they say like it's not important, it's only 1 sample out of 900, or they give the wrong denominator. Or you said it turned from B into A during culturing.
I didn't say that, by the way. Yeah, you said like reversion. That's not what I was saying. I was saying it's contamination. It's not that it turned into, it was contaminated. That's why it's so.
When did they sequence it though?
When they sequence it after culturing.
But that's like 2021, right? When Lineage A, they redid it in like 2021, and Lineage A is extinct by then.
No. They did the. Well, I have to double check, but I think they redid it. I mean, not redid it. They did it. They originally. Because I thought it's from culturing experiments, and they did the culturing experiments in January or February 2020. Very early.
Okay. And there's another, so yeah, they say it's fake. I pulled up some like Twitter, every objection to this, like Florin's a great guy on Twitter. He says it's Xi Zhengli's glove. And then you got, your colleague Stephen Quay, who says the sample is actually fake. And then there's like people found like the guy doesn't use gloves. He touches the seafood with his hands, and then somebody found a picture of his stall with some gloves in it. So maybe those are the gloves. And you said it's 1 out of 69, which is not correct because 69 weren't sampled. Or it's misleading. But Quay was even weirder. He says it's fake because he said like here's a regression for the cycle threshold for PCR and then the number of reads. And he said, with this cycle threshold, you shouldn't get the 1 million reads. And this was actually convincing for me at first, because a lot of these things are confusing enough to convince me until I look into them. That's the 4 samples that did the amplicon sequencing. So he's not comparing a fair,
but those are
same sequencing.
Those are the cultured samples. That's the thing.
Yeah. Cultured and amplicon, and maybe cultured. We're not sure about that one. It says they didn't do it. And then if you
culture them, of course you get,
if you do the apples to apples comparison where that's not the amplicon, the regression works just fine. So Quay's just pointed at the wrong misleading thing.
What do you mean? That's the problem. That if you actually use the CT, like the CT value for the 22 reads corresponds to this regression perfectly, but
but he didn't mention that it's a different sample. He didn't mention this different sequencing method. He just said it's fake.
Well, no,
they're lying.
I don't know exactly his analysis, but it's the same implication that the subsequent 10 million reads and the lineage A and the 2 or 3 extra mutations all come from like later process of either culturing or sequencing, and they represent contamination because that doesn't correspond to the CT, like the CT.
Yeah, so like he said the two mutations came from contamination, but I went through all the other first 800 genomes, and none of them had those 2 mutations. So it can't have come in the first February. It could come later, if they sequenced it later. But if you do it too late, then it can't be it can't have lineage A because that went extinct at some point in 2020. So you only have this tiny time window where you can find both and explain it all.
I think I saw at least one of them I saw in early genomes. I think that was actually on GISAID. I have to double check. It wasn't in GeneBank, but on GISAID I saw it. But anyways.
I'm just going to do a little bit of human inference for a second, and Sarah will be offended, but I think when you're looking for misinformation, it's a lot easier to figure out like who's a bad actor based on not what the science they're saying, because it's super confusing, but just like the way they present themselves. Okay, dude with a white coat and his microscope. And that's exactly what the ivermectin people did. Like Dr. Corey wears his white coat. Ryan Cole, he's looking at turbo cancer through his microscope right now. And you've got the hydroxychloroquine people that use their white coats as props. I started getting like Eastern European spam on my blog, and they'd send me their white coat protest, which was called the Frosty Silence of White Coats. I think sometimes you can tell like who's a bad actor just from the like pseudoscientist goal. You'll probably point up a picture of like
I think yeah.
Dude with the bow tie right now. I'm blanking. Peter Hotez.
Well,
Can
Peter
I get
Hotez,
offended on behalf
Bob
of
Gary,
someone?
Robert Gary. He's always appearing in his lab.
He did on Megyn Kelly, I think.
Yeah, so
Yeah.
He's
So
your,
this might not be a 100% rule, but that's my rule of thumb. We could probably go on to a different topic.
I just wanted to quickly ask, because you two talked amongst each other, and I think you left Will, at least you left me behind a little bit.
Yeah.
I know what PCR is, but what is a read count?
Oh, so they've like I had this table here, and they've sequenced it some way, and they've pulled up a certain number of, like, I guess a read is like you chop it up into 300 segment long bits of RNA or DNA, and then you Will could probably explain this better than I do, and then you hit a bunch of different sequences, and then you stitch those together to actually find the full genome of the virus. And so each time you hit a certain sequence in the genome, you're adding to the counter there. So here they've hit, 10 million. They've hit 29,000 bases on there, but they've repeatedly hit some of them.
I was going to say the genome I don't think is 10 million nucleotides long.
And so the reason why this correlation might make sense is because CT is a cycle threshold with PCR. You're like doubling it each time, and so then the number of reads should be like 2 to the n there.
Okay, so
And
it's highly redundant with the cycle threshold, which is the whole point of making such a graph. And then this SARS amplicon versus the other one, I did not realize there were these two different types of samples being done. Can you clarify what that is?
I think SARS amplicon is you do like a PCR process with a primer that's going to amplify certain segments. So you have this raw sample with a certain amount of RNA in it, whatever, and you're going to use a primer to amplify what you're looking for before you actually run it through the machine that counts the number of bases.
Okay.
And so you get a better signal out of that.
Okay. And maybe I'll retreat even further and ask, like, who took these samples? Where are these numbers coming from? Who did this work?
I mean, it was George Gao's group at first, and George Gao quit the Chinese CDC, and now he actually works on some like pandemic prevention group in the US a little bit, but it's William Liu is the new head of so it's Chinese CDC.
And these samples were taken very early, I assume, in
they're January 1st or January 12th usually.
Okay.
Yeah, January 12th are the raccoon dog samples. Like they went to the actual stall that sold raccoon dogs. Actually, the 2 wildlife stalls, and they collected I think 70 samples just from those. But the majority of the samples were done on January 1st. There was like 515 samples, out of which they got a bunch of positive ones. First they looked for PCR positivity, and whatever was positive by PCR, then they sequenced by NGS, and then they got the number of reads is basically out of whatever 200 million 43 nucleotide long reads, how many of those correspond to the SARS genome. And initially, most of the samples are like in the dozens or hundreds, but then they actually tried to culture samples, create like culture in a cell culture, put the viral sample, the environmental sample into culture, and see if they can then recover a viable virus, or like resequence that supernatant from those cell cultures. And some of them yielded, millions of reads. Basically, they were successful at recovering a viable virus. So yeah,
And they grew 3 of the viruses, I think, successful, but most of them they couldn't grow.
Yes. So they grew basically. When you see a repeat, like F13 appears several times. So if F13 is the code for the sample, like the actual swab from a particular position, right. And so F13
they cultured it and then maybe
F54,
passaged it again or something like that.
Yeah.
And one of those did mutate during passaging, but I think it was like the 2nd or 3rd generation. But like if A20 did mutate, that's like even worse for your case, because if it's A20, if it's Lineage A with no mutations, then that means the Lineage A root was at the market. Whereas if you're saying it's Lineage A plus 2 mutations, you can still pretend like maybe Lineage A came from the lab.
I mean, it's not that I'm saying it. It's fact that this A20 sample had at least 2 more mutations, maybe even 3, because the Christoph paper actually had.
But if those mutations are contamination, that's bad for your case.
Why?
Because if it's Lineage A with no mutations, that means Lineage A is more likely that it started at the market.
Not sure I follow. I'm not.
Well, like in your slides, you had one slide that said, "Hey, the first Lineage A case already had 1 mutation, so that means it must have been even earlier than we think." And now here it's like there's 2 mutations, so this is somewhat derivative of Lineage A, and it would be bad for the lab leak case if it was a pure Lineage A sample at the market, because that would confirm even more that Lineage A started at the market.
So you're saying it would be Lineage A, but the mutations are sequencing errors?
Well, you're saying they came from. I'm saying it's just Lineage A with these 2 mutations. That's the Occam's razor. That's just take the data as it is.
Okay.
But like Quay here is saying, oh, these 2 mutations came from contamination.
No, I don't think he's saying the 2 mutations came. The whole Lineage A came from. Doesn't matter.
He's saying all 4 mutations came from contamination.
Just the sample, the virion that yielded Lineage A after sequencing came from contamination.
So you need to figure out the date then if you want to make that case, to like exactly when it was sampled and how much Lineage A was going around, and is there enough to contaminate it?
And if we assume that this is a true sample with 2 additional mutations, then it's a later. Then it would have to be a later variant of Lineage A, which already accumulated by January 1st, accumulated 2 or maybe even 3 extra mutations. Which again, I think undermines the idea that Lineage B spilled over first, because you already have this Lineage A with extra mutations. But I mean, it's very hard to untangle using these dates which came first. I think phylogenetically, and you made a case that it's no way that Lineage B could have reverted to Lineage A, or like very unlikely, but yet I think kind of you're making the case that B spilled over first, even though it's phylogenetically later, and basically either everything we see in A represents, I guess, a reversion to the bat ancestor. Like it evolved from B spilled over first, and then evolved into A, and the A got amplified, or maybe it was 2 separate spillovers. And yet again, we only see evidence of. We don't like the evidence of A spillover in the market is minimal. Like just that Lineage A from that.
Well, there's the infected cage and carts and drains in that one shop. And those drains are positive well into February, when most of the market's not testing positive anymore. So that could be like, persistent fecal virus samples in the drains. And if it's just people sneezing into the drains, that should be everywhere, not just in the one shop that had the infected animals. So there is like a bullseye right on shop 629 in the market.
Well, I mean,
The only-
that shop, again, it had like, out of the 70 samples they took, only like 5 were positive in that shop, and they had like between 1 and 6 reads, six stars to reads, whereas like,
They took 10 on the first day. Like 70, you're inflating that by adding later days, I think. They took 10 in every shop on that day, every wildlife shop, and that's the shop that tested positive, plus like 1 in the freezer somewhere else.
no,
But
on
so it's
January
50.
12th, they took
Yeah, they took 10.
70 samples. I'm looking at the,
They took 70 across the market, but they took 10 in 629. They took 10 in every wildlife shop.
they only sampled on January 12th, they sampled like two wildlife shops.
No, they found 2 positive wildlife shops, but they sampled them all, so that's why it's unique. Or not all, they sampled like the 7 that were selling wildlife. You can pull up a map if you want.
I actually had a map as well, but we have a map.
Here, I'll share my screen again. So this is January 1st sampling. They sample like for all the human cases, and they find a bunch of positives scattered here and there. And then this is January 12th. They went for the wildlife selling shops, and so they found like was that 5 of them are negative, 10 each of those shops, and then this one 825 has 1 sample in the freezer, and then this is 629, which has 5 positive samples, and then it's got the drain samples later, and then they kept sampling through January to March, and the only places that are positive are 825, the one shop, and also this one is 0 reads now, but the drains are still positive for that one.
No, wait, I don't know. I have for January 12th, all the samples I have are in the. I don't know why you have so many. Oh,
I mean, we can pull up
the white circles, right?
yeah, these are negative. The white circles are negative samples, and these are
Yeah. So
know,
yeah,
I'll go to
basically
them
they
about
sampled only those stalls, and so they had only two positive, and most like 5 reads came, 5 samples with reads came from that orange one, right?
yeah, and then 1 was the freezer. So this is like the hedgehog shop, and that's like the raccoon dog
Yeah,
shop.
and basically all of the raccoon dog samples. Did they only have 10? I think
10 that day. They went back because they found the positive ones there, and they said you can tell. Like when they tested the animals, they tested the animals from those 2 shops.
But from the Bloom data set, which looked at the mitochondrial DNA reads from the raccoon dog stall, I think it had like 70 samples with raccoon dog mitochondrial DNA, and then only 5 had any SARS-2 reads, if I remember correctly, from like...
Yeah, so they were selling raccoon dogs in other shops too, is basically what that comes down to. That's what Jesse Bloom proved, that there were raccoon dogs in multiple shops, but they were only positive in one shop.
But
so that's the interesting question.
Shop,
is like, should they all?
Even in that one shop, they had many negative samples.
So this is the drain map also. So the drains in the market, they only test positive on the first time they go through, like 2 on the east side, 2 on the west side. 1 is right in front of shop 629 with the raccoon dogs. And then when they come back later, it's 629, and then it's only the drains downstream of 629 there. And I think SAR last week said that this was like raccoon dog, or no, mahjong room, but it's not downstream of the mahjong room. It's only downstream of the that shop.
No, I mean,
and
the
then
mahjong room is there with like the toilets. That's downstream.
The toilets are like right there, but these are overground. These are not like the toilet drainage. These are just the overground. It's these little gutters that wash out the shops, and so the waste from the raccoon dogs washed into that gutter and infected it. It's not like people pooped in that drain. to my knowledge.
Well, I mean, it's a drain system, and the aerosol.
And then when China tested the animals, you could tell they were interested in these shops. They were interested in, like, they went to the warehouses and they only went to the two shops that tested positive, 825 and 629. And so that's why they were interested in those shops, because they tested positive, and why they kept coming back. And when they tested the animals, we know which shops they went to. Like, we know they went to 629 and they tested lots of animals from that shop. And there's like one other.
Yeah, all of those.
Yeah,
animals.
but it's like frozen. I mean, we look at, they tested snakes, they tested like dead rabbits. So the only relevant animals are like, they tested 3 bamboo rats and 14 dead hedgehogs. They didn't test a single, raccoon dog. So it's hard to
Well, they also
conclude.
had like 15 raccoon dogs in Wuhan from vendors.
From Hubei, from a different part of Hubei, yeah.
Well, I don't know, different part of Hubei. I don't think we know from which part of Hubei.
This guy, it's right, we know because Tian published that. That's where they took them from. So they're, it's east of, and you probably really wanted to go like west to Enshi because that's where the caves are. But Tian wasn't allowed to go there. So, like, if 15 negative raccoon dogs proves it's not a raccoon dog, I don't think that's... It's like putting a cup in the ocean and, like, you didn't find a fish, and, like, oh, there's no fish in the ocean.
Oh,
Can I pull us out a little bit with 2 questions?
Yeah.
The first question, basically, I want basically both of you to speculate on how strong your case is, or to construct a case under the idea that I cannot conclude about your debate about these small number of reads and samples and things like that. Like, let's say that I don't assign either of you a winning position on which samples had how many coronaviruses in the first 10 samples that are taken in the market. Can you kind of help me through that? The reason is, it's, one, very confusing, and I apologize if this is just me being stupid, but I'm pretty confused about which things are where. And the second is, and this is from my background, Illumina reads, presumably these were done on Illumina, I don't know what else they would be done on, they all have barcode indices where you get to say which sample a read came from. And those barcodes are sometimes error correcting and sometimes not error correcting. But when you do the index read on an Illumina machine, it will make errors sometimes in those index reads. And if you don't have error correcting barcodes, they'll end up misassigning a read to a sample that it shouldn't be at. So there's all, like, my background is in bacterial metagenomics and bacterial genomics. This happens all the time. So I'm like real uncomfortable with being like, okay, there's 2 reads in this thing, and that tells me a lot. If you used an entire Illumina lane, you've used 200 million things. Even if you're using a 12 base pair Hammond code for your index read, which I don't think these would use, you still have a high probability of that. So what I'm trying to say is a lot of background evidence is telling me that there's a large error bar in this analysis, and I'm very confused by it. So with that, can you help me speculate or help me construct your case if I cannot understand this?
Who first?
Whichever of you wants to. Sorry.
Sorry, just wanted to make sure.
I'll go first. So you work with sequencing and I don't, so it's kind of hard to, like, I'm a consumer of, the only COVID test I can use is the kind I put up my nose. And, like, I had COVID and a fever of 104 and my test was still negative on the second day, and then on the third day my test tested positive. So that's my idea of how accurate testing is. And now they can come 12 days after the market shut and probably like 6 weeks after the spillover happened and tell you which shops were positive and which animals were in those shops. That's like beyond science. That's kind of magic to me. And then the objection is there's not enough reads in there, so let's toss out the whole zoonotic origin, and maybe it's the lab instead, even though there's like no evidence for the lab other than like the diffuse grant sounds sort of like this virus if you squint at it just right. I'd like for it to be more reads, I guess, to make my case stronger. I don't think that throws out the whole natural origin.
I'm not saying that. I'm saying, like, basically say that if you exclude any sort of analysis, like any of the data that you just showed for which lineage was where in what proportion from, let's say, December 1st to January 30th, how does your analysis change? Like can you still win this debate if I can't understand this?
I have no idea if I can win the debate if you can't understand it.
Okay.
And I tried to pick really smart judges to avoid that. But I would not do this with two random people. And I think you guys are both really sharp. And I think we can work out the details with written questions and verbal questions. If you're asking, like, can we throw out a certain amount of data and still have my case be strong? I think, like, we could throw out the glove sample, the A20, and you still know that Lineage A is associated with the market because the first two Lineage A cases were, lived so close to the market. And I think, like, with the Bayesian analysis on the molecular clock, sorry, too many big words there. You can throw out all the market cases, and the molecular clock is still broken. Like, even if you exclude all the market cases, with them it's like base factor 48 that B came before A, but without them it's still like base factor 11. And my analysis uses the 11 just because I wanted to say, like, hey, even if there's sampling bias, B still came before A, and that's really hard to explain with the lab leak, but it's easy to explain with the two origins. So yeah, I think you can take out certain parts of my analysis and it's still true.
Yeah, the goal here is not for us to dodge the work, but I think both Eric and I, like, your facility with like, you guys have already committed to memory all of these different sample names and all of these different things. And, like, I'm going to have to review this video several times. So I'm
we can give you more weeks also. Right now you have 2 weeks and it's going to be over Thanksgiving. And if you're going to be in, like, a turkey coma while you're supposed to evaluate this evidence, I'm happy giving you another week to parse it or another week for written. But, I mean, what other things would you want to exclude? Like,
let me hear just Yuri. What's your kind of so in a similar way, what Peter constructed there was, like, let's say his Bayes factor is 11 versus 40 for lineage B being first based on the PRRAR model and the C to T transition, I think, in there. Is that right? Or something like that. What's your case for lineage A being second or being first?
Well, I mean,
if I don't move the market,
if even, like, my only interest in lineages B or A, which was first and which wasn't, is only in relation to zoonosis making the claim that they spilled over the market. If you exclude the market, where it's, as I think you're rightfully pointing out, very noisy data, if you exclude the market, I think the case for the lab leak is, doesn't care if B came first or A came first. Basically, the market is the only thing that is, as I said, it's the kind of the linchpin of zoonosis. So if you exclude it, I mean, there's no case for zoonosis, period. And even if you include it, like, it's a very weak argument to say that just because we see the two earliest lineage A genomes somewhere in the close vicinity, close proximity of the market, they must have come from the market, although we don't see any patients with lineage A with a link to the market. And under lab leak, it's still, like, completely plausible that lineage A, like, lineages were circulating in the vicinity of the market within, like, the population center of Wuhan, and one lineage B got into the market, got amplified there. So, but I'm kind of going off on a tangent. So basically, that said, to make the case for the lab leak, you don't really need the market, or basically, yeah, like, if you exclude the market, the case for zoonosis is even stronger, because the market is the only thing, essentially, to my understanding, that reduces the probability of the lab leak.
Okay, that's helpful. I think to answer the question that I cut you off from, Peter, I'll certainly be constructing, like, my understanding of each of your cases before the next debate and sharing those as part of the questions to make sure that my internal mental model is not, like, I'm not doing a disservice by misunderstanding pieces. So it will come out.
Yeah, that's
Peter should have
great.
about another minute because, well, you were talking and running on Peter's time.
Sorry. I'm putting my foot on the scales, but yeah, I apologize.
Oh, sorry. Also, I was pointing out with the two cases close to the market, it is actually a pretty good coincidence. Like Warby made a null model where just like you pick a random address in Wuhan based on the population density, and it was like a 3% chance that it would be, 2 kilometers from the market if you pick one at random. It's like, if you get a deck of cards and you say, I'm just gonna shuffle these, I'm gonna pull the Queen of Spades out, that's a 2% chance, right? Like 1 in 52. And then you do that twice with the two cases, and you shuffle the cards again, and you pull the Queen of Spades again. Like it doesn't sound like a big thing, but it is. If you're saying this is really the lab and they're really that close, you either need to prove that they're biased, or you need to explain how that happened by chance. And one of them is not biased because it was the first case found by what's her name, Zheng Yishan, the first doctor that discovered COVID in the connection, and she found this case before her market patients. So she didn't know about the connection. That would just happen to be that person lived that close. So it is a really big coincidence, and that's one of the factors in my probabilistic analysis.
One was already because of the connection to the market.
The second one's the hotel, and maybe that's biased. I don't know how that was, so maybe you can toss that if you want to, sensitivity analysis, pull that out of my. But like also, we've had all these excuses for why it's biased, and none of them have held up so far. We talked about this in the first debate where we're saying, oh, they all got diagnosed, before January 18th, when we hadn't removed that criterion from the diagnosis. But then I proved that most of them were diagnosed after January 18th. So there could be some reason for bias, but it's not that reason. And I also showed that there was, before December 29th, before the connection was known, it was like 60% market. So there's no bias there. So you can't just, that's really frustrated me in the first debate, where SAR can just say that data is biased, that data is fake.
How was it 60%?
But then I have to put the work in to show that it's not the case.
I think it was 33% market linked to QAnon. There's 55% with some link to the market, including markets that didn't sell wildlife, but only 33% of the early cases had a connection to the Hunan seafood market. And also, I think there is even
the December case, the full December cases are 33%, but I'm talking about before the bias was known. So before December 29th. This is Warby's 2021 paper, dissecting the earliest cases of in Wuhan or whatever, where he went through the early hospitals and said, how many did they find before there was that bias? And before that bias was found, it was like 50, 60% were market linked. So that's really hard to explain with a lab leak.
I'm not sure I remember that exact number from Warby, but I do remember the 33% with a link to the market out of the early patients, like December.
Sorry, I don't mean to rehash everything we've talked about already, but
I think recontextualizing previous things is helpful.
okay. So is the data biased? Yeah, so this is what I pulled up. And so this is December 31st. They found the connection on like December 29th. And so it's like 62% at that point. And there's this period after that where they are looking for the market, so it gets a little more biased towards the market. And then to January 22nd, it's 50-50. And then after January 18th, which is after they've removed the market link in their search, it's they're starting to do a retrospective search, and they're finding mostly not market linked at that point, but they shouldn't be biased at that point because the market link is removed. And so then when you look at Warby's diagram, the famous bullseye, all these blue cases here, most of these blue cases were found after January 18th, after the link is gone. So those are the retrospective search, and they're still really close to the market, way closer than you expect by chance.
But I think they were diagnosed before, if I remember correctly. And the 61%, so these are just the 16 first cases with a link to the market, the Lineage B cases. And I think they only looked at that hospital right next to the market, right?
No, I'm not talking about in the I'm not talking about, you should have watched the first debate, or I should have had the first debate with you. This is like, at the beginning, this is like, what's her name, the first patient, Wei Guizhan. So she gets sick December 10th, and then she goes to the hospital, and she's in like, not like a coma, but she's just like completely out of it for COVID. And when she comes to, there's other Huanan patients at one of these hospitals, and then they're saying at Tongji, there's also patients at the other hospital. And then you can go to other hospitals and look at their early patients also. And there's like 5 different hospitals that have a substantial majority of their early patients are from the market. And so like Warby looked at the case search in one of those markets, was Dr. Xing Yishan. And so she found like these first 2 patients who had pneumonia of unknown etiology, and she ordered the lung scans because they looked like SARS, not because they were from the market. And this is actually the first Lineage A case that lived super close to the market, but she didn't know that yet. That was only discovered retrospectively. And so, and then after that, she got 4 more COVID patients that were linked to the market, and then she talked to her colleagues at the other hospitals, and they were like, hey, this is weird, I have market-linked patients too. So at that point, I think you have like 14 and half of them. And then if you go to Zhongnan, which is right next to the Wuhan Institute of Virology, December 31st, they say, hey, do we have any of these cases? And the 2 they find are both market linked. And so, and you add all those numbers up, and then you get the, and I think when I did the 62%, I added these also. But even if you remove these and just look at the early hospitals. So these aren't necessarily market patients. It's just like every hospital in town, even the one right by the lab, is having like 50% market cases before we know the market link.
Yeah, in terms of
I think
that 61%, like this first 16 patients all being lineage B, and so she found the first lineage A patient, I think the symptom onset was claimed. That's a cluster, right? A family, husband, wife.
Yeah, that's the one I was talking about here with Jing Zhishan. So that's the 63-year-old man and woman.
Yeah, so
these are lineage A, and their son also.
They didn't have a link to the market. And
right, but they lived 2 kilometers away. So this is the earliest lineage A that we're talking about, and we're saying like, why is this so close? Because it's unbiased, because this is before anyone knows about the link, and it just happens to be right in that same neighborhood that had the market, that had lineage A, lineage B, that had the A20 glove sample.
this actually helps, I think for me, this is the kind of analysis where this is somewhat abstracted from the specific read counts. So this is helping me understand what it looks like if I don't, use that read count things. But I'd like to focus on 2 other questions I had. Eric, do you have other things that you want to get in on this piece, or do you guys want to go back and forth on this piece more?
I'm ready to move on from
I just
market discussion.
wanted to like point out that
But we can let Yuri talk.
regardless of like what we think the criteria for early sampling were, I think we do have evidence of a sampling bias if we look retrospectively at this seropositivity in March versus seropositivity in January, and see that in March it was much lower for like the market district than originally, like the sampling. And I think, yeah, the initial sampling is, they have many more cases in the early sampling data overrepresented. And actually this
This is what you're talking about, or this even shows it better. So Yuri's saying like the 2 adjacent districts from the market are slightly higher prevalence than
slightly twice as high.
in this one it's, yeah, and this, let's see, this study, it's 5% versus 6.5%. So no, that's not twice as
Well,
high.
this is
And
April.
also
The other one is March, so the earlier.
I don't know which month this one is. But I think you're really missing the forest for the trees here if you're like, oh, I'm worried that this is 6 versus 5, but you're not looking at, hey, in the lab district is way lower. Like, we know which side of the river it's on. We know that it's concentrated
We're
there.
not claiming the lab, like this is I think the mistake that Rasmussen and Warby say that, if it was a lab leak, you'd have everything centered around the lab leak. And a lab leak is not like the market, it's not like people coming and going and getting infected. A lab is just people working there, and it's actually kind of in the more remote part of town. So if somebody got infected by the lab...
No, it's right in the middle of town right there in Wuchang.
Well, I mean, more remote than where the Hunan seafood market, which is like at the center of, well, close to the center of the population density. So I don't think why anybody would expect the district around the lab to have the highest seroprevalence rather than some, place with the highest population density.
So when they look at patient Chen, they point out how close he lives to the December 8th, but actually 16th guy. They point out that he lives really close to the lab. So that's like inconsistency where when you do find somebody that's close to the lab, you say, oh, it must be a lab leak. And when you find it centered on the market, you say, oh, well, this style of analysis doesn't work at all.
Well, I'm not pointing to patient Chan. I'm just saying, well, in terms of, the point I was making in terms of seropositivity, that the earliest analysis had like half of the cases come from the market district and very few from adjacent districts, but in terms of seropositivity, the market district had a much lower seropositivity than adjacent districts, which I think to me shows...
No, much lower. It's 5% versus 6% here.
Well, this is April. Like if you look at the March data, and I had a table, I think even SARS had that table in the presentation.
So you think people got unsick between April and March, or what?
Seropositivity falls with time, right? You have antibodies wane as...
Okay, so then they're lower in the market district because those cases were earlier and the antibodies have waned more.
No, they couldn't have waned like twice as much. And Wuhan went into lockdown, so basically everything closed, I think, January 23rd, right? So you have a snapshot of that data. So, and of course, you could have a difference between sampling methodologies between the March and the April study. But, well, to me, this is evidence of a sampling bias early on in the pandemic centering on that district where the market was.
I mean, the other thing that came up in the first debate was we looked at Shinfati market and we found that the case, where it was like imported seafood and that spread outside of the market. And I pulled up a paper there and the cases spread out from the market, exactly the same as Huanan. So we have confirmation that that's what happens.
The Shifaldi market, is it confirmed that it was like actually frozen salmon? Because at the time it sounded very...
I mean, it tested positive and it tested positive in the warehouses also. So there is China could be lying about that because they want to maintain their zero COVID illusion or something, and they could say it has to come from somewhere outside of China. But if you take the evidence at face value, I mean, if you believe it. But we do know it spread out from the market.
But, well, I mean, I think this is an important point because if it was brought into the market by an infected patient and then radiated from the market, then it's this exactly same scenario as a lab leak versus, a live animal.
Sorry, that's my first. Like this was the cases outside Shinfati.
And, yep, I think, Shinfaldi...
They're pulled out on the market again.
Shinfaldi,
and then, you center them.
Kind of maybe just strengthening the idea that super spreading location would radiate like a market, like basically market being a perfect super spreading location, it would radiate. Then you could have
right because they have infected animal products that get shipped into them, and then they infect people.
Or a human brought it into the market and infected people. It's not like.
But this is zero COVID in Beijing, so it makes sense that the source from outside of Beijing can ship the infected thing in, and then it spreads outside from the market. Whereas it doesn't make sense. Also, I think when we look at the phylogeny, we can actually see that this is frozen. Like the strain is an older strain from the thing that's currently circulating. So you can actually kind of prove that way.
Was it a European strain, like a strain circulating in Europe at the time?
Yeah, I actually have that, I
But
think.
like I think regardless of that, I think it's still an example. Like we can't differentiate between a lab leak bringing like a frozen sample into the market or
So
an
here,
animal.
this is Shin body, and we have these are like months of the samples, and so we're building. I say it's single polytomy, right,
Hmm.
from single introduction. But then notice the months here, like it's these are May, and then these are April. So it's more closely related to the April strains than it is to the May strains. So that's another piece of evidence saying it's a frozen sample because it's like a little bit, behind of what's currently circulating in Europe or wherever it got shipped from. So that
But is it a
yeah, you
European
don't
frozen sample?
necessarily,
Because it could have been just
yeah,
like some sort of delayed reinfection by a local person, and we wouldn't like, and like yeah. Regardless, if it was a frozen sample, I don't think it's similar to like an infected animal spreading it to multiple people. It's still like a single point, and the vendor selling this fish getting sick, and then spreading it to the market. So I don't know if you can differentiate like this is how raccoon dog infection would be versus this is how a lab leak super spreading by the market would be.
sorry, I was looking for a slide. I wasn't listening for a second there.
Yeah. I was just saying that I think the example of that Shinfaldi market can basically serves that we can differentiate between a lab leak bringing a infection into the market and the market then amplifying it, and actual live animal bringing an infection to the market and the market then amplifying it.
How is that different though? It's a frozen infected product versus a live infected product coming into a market and then spreading.
No, I'm just saying that it's the pattern is the same. Like if the market is amplifying things, the market kind of doesn't care what. So you can't using that pattern of like spread, the concentric spread. You can't make a dispositive argument that this must have come from zoonosis. Basically,
But lots of things amplify. There's super spreaders at churches and bars and restaurants and any crowded indoor place. This is an argument in the first debate. Like there's this mahjong room at the market, but there's also like 3,000 mahjong rooms or similar social places in Wuhan. So any of those could have been the first super spreader. Why did the lab virus go straight across town to the market and spread in that
well, I
mahjong
think the
room?
market has a very nice split between like persistent population of vendors that can, and people going through it, and the vendors can like they always interact versus a church. You get into this church for like a couple hours, and then you really need to have, I think, already a persistent virus to get amplified well by a church. But yeah, we're like this is the conjecture land. Like why is the market better? And of course, animals like again like raccoon dogs. If they're so good at spreading SARS-2, having them in the market would be also a very good conduit for amplifying a lab leak virus that you bring to the market. Then,
Raccoon dogs would amplify a lab leak?
well, why wouldn't they? Like if
Well, because then it has to be reverse zoonosis and then forward zoonosis again. And half of lab leak Twitter says they don't even believe raccoon dogs can be infected because they don't shed enough virions. And now you have to throw out that argument just to.
half of the zoonosis, or 100% of zoonosis, say raccoon dogs are the most likely source. So if raccoon dogs are so good, and we didn't even get to the raccoon dog experiment. I have it. It was in the deck. There was only like 6 of 9 that got infected.
That sounds interesting. What's this raccoon dog experiment? I'd like to hear it.
Oh, the raccoon dog experiment is they try to
Do you want to share that or?
ascertain. I can just speak to it.
Yeah, can you share a slide?
I mean,
Or you can just
I
describe
would
it.
spend more time looking for that slide, I think, than just describe it. So yeah, basically they try to ascertain how good are raccoon dogs at spreading SARS-CoV-2, and they did an infectivity experiment, kind of like the ferrets with the H5N1 all the way back, the original gain of function study. Basically, they got a bunch of raccoon dogs, and they tried to like in adjacent cages. They had like 9 raccoon dogs, And I think in every cage they infected one, and they had 2 other ones, if I remember correctly. And then they used, I think, a very high titer of the virus to infect raccoon dogs, and only 6 of the 9 raccoon dogs actually got sick. And also they had like a 10,000 or 100,000 lower titer than seen in human infections. And also they actually used a later strain. They used a strain with that D614G mutation to do these experiments versus like the earliest strain that would have been circulating in the Wuhan or the animal reservoir. So it's still kind of not conclusive that raccoon dogs are all that good at first of all transmitting the virus among themselves, and they didn't get like visible signs of being sick. So in terms of like how much virus were they shedding, and was it enough to infect humans? I think.
Do you find this experiment, which is much less exciting than I was expecting from the words raccoon dog experiment, do you find this experiment like persuasive in thinking that this means that raccoon dog could not have been an intermediary step in a zoonotic transmission, or are you happy to be ambivalent about the experiment having any meaning or significance?
That's for Yuri.
That's a question for you.
Oh, for me. Oh, I'm sorry. I thought you.
Yeah,
asked
like,
me.
are you persuaded by this experiment? Or is it just something someone did and maybe they messed up?
No, I'm not persuaded at all. Yeah, I don't think, we have strong evidence that raccoon dogs are good at transmitting the virus, like the actual live virus, because we didn't even try, even with the later version, the more infectious version of the strain, that was like 3 times more effective, if you remember the Mark Eluar experiments, where they used actually like to try to see the difference between the original D614 strain versus a G614 strain. So there was a 3-fold difference in infectivity. So even with the latest...
What I'm asking is are you persuaded from this experiment that they're bad at it? Because, like, do you think this means raccoon dogs cannot be an intermediary host?
No. I don't think that completely rules out the idea. But I think it weakens this notion that... Yeah, raccoon.
Well, but they are really good for amplifying a lab leak. They're just not very good at spreading a natural virus. I think that's what we've established.
No, I mean, if this is the conjecture, or no, if this is the point you make, that raccoon dogs were very good at transmitting the virus and they brought the virus to the market, that under this very same kind of prior of raccoon dog being so good at transmitting the virus, they could also be very good at amplifying a virus brought in to the market externally.
But they're equally good as humans, or assume they're equally good as humans, and now you have 10 million people in Wuhan plus, 100 raccoon dogs or 40 raccoon dogs or whatever it is. And why is that extra 40 any different than 40 more humans?
Well, I guess if you have all the vendors kind of representing raccoon dogs, and humans are, I think, better at transmitting the virus, but if you have, I guess, infected raccoon dogs at the market, you couldn't make an inference whether they were infected just by seeing, like, again, collocation of raccoon dog mitochondrial DNA and positive samples from that stall. You couldn't infer whether they were the original animals who brought the virus to the market, or if it was infected raccoon dogs, whether they were infected in the market rather than they were infected outside the market.
I think we came up to this question in the last debate, like, how many patients do you think there were before the market outbreak?
How many patients there were where?
COVID cases in Wuhan or the world or whatever. You think, like, there's this big outbreak before the market. How many cases were in Wuhan before the market outbreak?
I have no idea. Like, you're asking me based on the current observations. Well, I mean, first of all, we don't even know. Like, we haven't sampled enough early cases to conclusively say like we have a huge confidence interval, even for the kind of spillover date, like the Warby models, it's like put it between October and November. So with such a wide interval.
But they also built a model of like how many people how many cumulative cases there could have been based on that genetic diversity. Because like we agree there's such low genetic diversity, and that means either it's a lab leak that just happened or like we just caught the spillover. Like it hasn't been hanging out for 6 months. So have you thought about like how many lab cases there were before the market?
Not 6 months. Well, I mean, we don't even know how many market cases there were. We just have kind of those 16 patients in the hospital. How many were asymptomatic? How many were symptomatic but didn't go to the hospital? So I don't know. I mean, at this point, this is just pretty useless speculation. I'm saying.
Or do you have a guess for like when the lab leak happened? Have you thought about that?
Sorry, we need to pause. I just accidentally reset the time for you, Peter. I think you were at 20 minutes. You guys?
Yeah, you can take 11 off. I don't care as much as SAR about the equal.
It was close to that. I apologize. That's my mistake. It's hard to switch this thing.
It's going to be merciful when the timer hits 0, so don't.
Sorry.
Where were we?
Yeah, well, I don't know, like in terms of cases, and in terms of the data, the lab leak probably, and yeah, if it's like cryptic transmission, it could go on for pretty much, for weeks, if not months before it blows up, right? So, I don't know.
I don't think that's true,
October,
and
November,
I think that,
well, and also, yeah, in terms of like mutations, if it had any prior mutations that were like made it less transmissible, which we have no idea about. I don't know, but
Can I use this point to ask a question to focus it? I've talked a lot, so Eric, if you want the first crack, but I want to switch.
You want to move on?
Can I just say something? I'm actually just going to fill in the time here for a sec before you guys get to it, because we talked a lot about this at the first debate. Like I presented this exponential model, and I said like you can't have 1,000 cases in Wuhan because if you did, you'd have too many deaths by the time the lockdown happens. And I'm not going to repeat that again. You can watch the debate again. But I looked into this a little more, and you've got this. Another thing I've learned is that COVID transmission is over dispersed, is what we say. It's not like you say R naught equals 2.5 or whatever. So that means the average person infects 2.5 other people. But it's not actually each person infects 2.5. It's like most people infect 0. These are some guesses on the COVID parameters. Most people infect 65% of people infect nobody. And then a few people infect, one person. But then there's this long tail of people that super spread. And so this is like, this is what you expect it looks like, where it's like everybody infects 2 to 3, but it's actually this different distribution, this negative binomial. And so there are interesting things you can understand about this. Like it can spread faster than you think at the beginning. That's the orange line versus like the red is what you expect from the Poisson, I think, because it can super spread right at the beginning. But this also explains like why it goes extinct so often and why like this can't spread outside of a city. Like if it spreads on a farm, it's going to be that case where it just goes extinct right away. And so the way that it does spread is like either it goes from the raccoon dog to the person and it just stops after that, or it goes from the raccoon dog to the person and you get the like mini super spreader that starts that exponential chain. And that's like why the PRRAR2 spillovers make sense, like because like 2 of those stuck and the others didn't stick. But this is why like it's just floating around. It came from the lab and it's going one person to one person to one person until it finally hits the market where it has the perfect conditions to spread. That's really unlikely with this kind of model because one transmission with no super spreaders just doesn't sustain COVID. You need a super spreader somewhere pretty fast. And if it's the lab, like the first super spreader is probably going to be at the lab and all the smiling people at the dinner table are all sick already. I mean, maybe that's why they're out to dinner, because they all know they're immune already. But if it's not the lab, like there's so many other places it could be they could go to, like their favorite restaurant, or another market that's closer to the lab, because there's like a bigger market that's on the same side of town, or a supermarket or a restaurant or whatever. So that's like, it has to hit the market and hit this first super spreader like within the first 20 cases or whatever. So even if the market does have these perfect conditions with the resident population and the mahjong room and all this stuff, even if for some reason it did spread the virus better, it's still unlikely that the first person before it goes extinct or super spreads is going to reach the market. And this is just an interesting model that explains a lot of things. Like, it maybe explains why Japan succeeded with COVID, because they went after clusters rather than our like stupid lockdowns where like you shouldn't talk to your friends, but you could still go to a sports game or whatever. And then it explains why it starts in a big city, and again it explains why you can't really get this like stuttering person-to-person transmission. A yield.
Yuri, did you want to respond there?
Yeah, well, I mean, I think it's also, there are plausible scenarios under the lab leak where it could have been, it went extinct a few times from, people infected in the lab, but of course the lab was this persistent infecting, space that infected either novel people or reinfected people, and then they actually met that lucky super spreader that eventually, caused it to spread. So it's just very hard to speculate on the exact number of patients early and the exact dynamics before it could have been amplified by the seafood market. So, but anyways,
I don't know. We don't know, but just like every time you have a lab leak, there's the chance of that super spreader at the lab and all these other places. So even if you're like, there's multiple lab leaks, I don't know if that makes it more likely that it's. And I still don't see why the market's like this perfect place in town that's the only place COVID can spread. Maybe we can talk about that more with SAR in week 3, because that was his idea more than yours. Sure. Maybe let's just talk to the judges about their questions instead.
I want to know if the two of you want to talk about N-glycan at all. That was my question.
I already talked about it. If you guys have questions about it.
Okay.
I mean, you did talk about it, so I guess I'm asking Peter if you wanted to give a response to Yuri's claims about N-glycan.
I mean, I sent you guys a written response already. I can read that on the screen if you guys want. I think it's probably Texas sharpshooter fallacy, because there's like 22 different N-glycans that it can be, right? It's not just there's these 2 mutations within the SARS virus, the original SARS virus, and it was the the 227 and 699. Yeah, I got those right. And so if they had manipulated those 2 sites here, like, I could see a stronger case for it, because that's the ones they'd be interested in. But then it's actually this like 370 site, and then I looked over all the sarbecoviruses, and that site does vary. It's about 10% of the other sarbecoviruses have a mutation there, so it's possible. But also, you might say, okay, it's 10% chance it's natural, 90% it's a lab leak, but that one is not special, because there's all these different papers that say like 370 is important, 330 is important, or here's like 7 different sites that are all important. So you could make a theory for every one of these. And also the diffuse grant says they're going to remove glycans and they're also going to add glycans. So you have 2 different theories there. So the odds that you're going to get one of those theories pop positive, especially, we also know that these change if it does jump species. So maybe that's just a sign that it jumped species. I think it's the same with like the FAO-I site that we were looking at. There's like 3,000 different restriction sites. I sent some slides about that also. Yuri's found this one that's significant in the FCS, but like he found the FAO-1, but here's like every other restriction site I found there. So why is that one important and not the others?
This is a bad diagram. They use 4 nucleotide long, and it's ridiculous. The Panchin and Tishkovsky, that's the paper. But
well, and they said, you said it has to be like 5, and then they said if it's any one, it's 99.5%, but if it's a longer one, it's still 50%. You're going to find one. And then you found two papers that worked with this, and I wrote it down while you were talking. It's like, it's not coronaviruses. It's just really, it's like a salmonella experiment, and then like a strawberry model virus. So like, yeah, maybe the strawberry model virus people were working with Xi Jinping, but maybe you just found a pattern and then you tried to explain the pattern.
Well, no, I just found this particular site in this particular insertion, and this is the hypothesis I formed, and it could have been just, by coincidence that it arose in the insertion, or if it wasn't a coincidence, this is why it could have been used. And I guess for the glycan, it's the same thing. Yes, I mean, obviously anything can also be natural. It could have lost the glycan naturally. It's just this coincidences keep building up that it's the same glycan as the diffuse cited in connection to this DC sign binding with the macrophages, and they were interested in a whole bunch of glycans. So it's not like they had to zero in on one particular one. It could have been one of many that they manipulated, and it just happened that this one really helps with the infectivity of human cells, and that's why this particular strain maybe jumped out and infected someone in the lab. But
But it's also like they're gonna make all. If you imagine they're doing diffuse, they're making all these different viruses, and you're imagining they make every experiment all at once. So like they could have made the WIV1 backbone with this special RBD, and that would have leaked first because it's good enough. And then they put in the furin cleavage site, and then they put in the N-glycans, and none of those leaked until they did every experiment at the same time. So you're imagining this huge body of work that's happened before any one of them leaked.
Nothing has to leak. I mean, they're supposed to not leak, right? They're doing these experiments.
Yeah.
I'd like to focus on that, because that's where one of my questions was about this, is the sequence of events in terms of this mutation, Yuri. So how does in the lab leak scenario, how does the strain that leaks end up with the ablated glycosylation site and the FCS inserted? Is it one then the other? They do them at the same time in the experiment? How does that look?
Yeah, probably if they had a virus in which they inserted the furin cleavage site, they could also, in other experiments, see that this glycan is the one that really increases infectivity, and then they try to combine them to create like the mother of all coronaviruses, the most infection variant. Like they say in the diffuse proposal that they're going to have a mix of the mutations in the quasi-species to see how they affect jumping from species to species. So they could infer each of these things impact on infectivity separately, and then they can combine them, like that polymutant virus that I mentioned in the omicron connection, where they combined all the mutations into one strain and show like, oh, look how good this is at infecting humans. This could have been similar inspiration for them to do in the context of diffuse-like research. Okay, let's try to see what's the most dangerous virus that can emerge from that cave through some sort of natural evolution or jumping into some intermediate hosts, and and then have the potential to be really bad for humans. So that's my kind of.
Shall I ask a follow-up on that?
Or as long as we're on here, I just wanted to know what Yuri thinks of his former coworkers' current theories. Do you have a preference on Fort Detrick versus the Wuhan Institute of Virology, or?
It's a loaded question because you probably seen the tweets, so you know my opinion, right?
I don't remember your response there. Sorry.
I should. No, I don't think Valley or Fort Detrick. I don't think Valley is COVID. That vaping disease, I don't think it's COVID, and I don't think Fort Detrick played any role in COVID.
So I'm looking at the diffuse proposal. In the very beginning, it talks about, we'll sequence, blah, 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 insert them into bat, SARS-related coronavirus, WIV1, STH14 backbones. And it says, these are not SARS and are exempt from dual use and gain of function concerns. I'm just sort of wondering, Yuri, if you had any comments, because as we discussed before, obviously the diffuse proposal is not a blueprint that everyone followed identically. In nobody's theories thinks that happened. But it sounds to me like the authors of this proposal are aware of, the potential of a risk of creating dangerous virus. They're talking about using a virus that they think will not be infectious in using a backbone they think will be not infectious in humans. I guess clearly something happened differently from their intention of not doing something dangerous and the theorized lab leak. And so just want to understand your perspective on how is it they did something dangerous by accident when, clearly they're aware of the need for safety of some capacity.
Yeah, no, that passage about them being exempt.
And I don't just mean lab safety in terms of like they have to wear masks, but also just general idea of don't do something dangerous. But sorry.
Yeah. So, but no, like that passage in diffuse, it's essentially just like a cover your ass passage to alert DARPA that this is not like gain of function research under the pause for gain of function research, because they had some exemptions that like bad coronaviruses, they can be worked and they didn't constitute this P3CO, like the dangerous pathogens gain of function research that was kind of banned. And of course, they basically said that we can do this research in our BSL-3 labs, and the fact that it's safe because it's I guess bat strains and it's not exactly SARS-1. I think under some gain of function definitions, like SARS-1 was not, you couldn't even do the experiments with SARS-1. I think in UNC you had to do BSL-3 plus for SARS-1, but bat coronaviruses you could do. And the same thing in Wuhan. They actually allowed them to work under BSL-2 on bat coronaviruses, but SARS-1 was a higher level of biosafety. Sorry, yeah. So I'm sure like they recognize that if they splice in a spike that can has good affinity to a human receptor, there's always possibility of it spilling out. I wouldn't spell it in the grant, I guess. We're going to create pandemic pathogens.
So when Peter said that it would be standard practice for many people in this field to work with pseudoviruses that could never leak, is my understanding, in this way, you're saying that presumably they'll be aware of this technique of using pseudoviruses and they chose not to use it because it's not appropriate for their research goals or something like that. Yeah,
and they felt safe enough using chimeric SARS viruses because it's going to be done in the WIV. This is like repeated twice, and it'll stay in China. US will be safe. WIV will do the live virus work. But they could have done the pseudovirus work, but of course it's very limited. Like pathogenicity in mice. So like chimeric viruses was their first step, and of course those are live actual viruses.
Okay, maybe Will's questions? Or?
Yeah, I think I'm running out of questions, and certainly out of brain power, but this is an important one that I think for Peter, I think I kind of want to understand what the chain of transmission is in your model, like how the zoonotic outbreak works. And I think it's been sharpened a little bit by what's happened in this debate, because it seems like if the raccoon dog is not actually very good at transmitting, I'm not saying that we've agreed on that or anything like that, but if it's not very good at transmitting, and we all agree that the virus couldn't have been circulating for very long, what does it look like? It seems like it has to get out of its animal host pretty fast into the intermediate, and then pretty fast then into humans to both not pick up diagnostic mutations or mutations that we would see, and spread in the pattern we have. Is that a fair characterization, an unfair? Speak to that.
I don't know that it transmits poorly in raccoon dogs. I mean, it transmitted to 6 out of 9 of the animals, and that was good enough. And also they weren't very sick, so that could actually be a plus for maybe they wouldn't transmit or, traffic the animals if they were visibly sick. And also that experiment showed no characteristic mutations like they had with the mink or whatever. So it confirmed in my mind they're one of the plausible hosts. Though, I'm saying it's like 99+% that it's the market, but I'm not saying it's like 99+% that it's the raccoon dog. I think we need to do more experiments on those animals to figure out which animals are most likely. Sorry, I'm running out of brain power too. What was the next step?
No, that's. I mean, that's where I want to go. So
it doesn't have to be really good transmission. It just needs to be good enough to transmit to people.
But for the overdispersed, the overdispersed nature of kind of like transmission events, I think that's what I'm kind of what I'm trying to kind of figure out is presumably there's a bat in the wild that has this under zoonosis.
Yeah.
Is consistently getting out of that bat into an intermediate host, perhaps raccoon dog, perhaps something else, and then those are continuously not spreading it. One makes it to the market and then gets one and then is one of the randomly rolls a 20 spread there. Is that the
yes, something like that, and it does that twice at the market, and it does it like nowhere else that we know of. But I mean, I think I put this in my supplemental slides last time. Like, it's hard to compare SARS and COVID because they shut down the country with SARS.
Just in terms of sequencing evidence or terms of intermediate sequencing evidence kind of thing?
No, because Yuri and SAR both said the same thing, where it's like, why is SARS in so many different towns? And, sorry.
Yeah, I know what you're talking. Yeah, I know what you're talking about. Yeah.
I mean, I don't know if you looked through all my slides last time, and
Yeah, this is the question of shutdown and which samples were actually sequenced, which were not, and where could you actually have seen the spread given the lockdowns?
Yeah, I'll pull it up if I have time, but it's like, if you shut down the country in 2 months, and if you shut down the country in 2 months for SARS, it's only going to hit 2 towns. And if you shut down the country for COVID, it's only going to hit 1 town. So there wouldn't be enough time for either of them. Railway might actually, okay. Here's what I was looking for. So this was, Yuri's diagram, and he's like, why is SARS in all these different towns, and why is COVID only in one place? Well, so if you mark the market closed and the Wuhan lockdown onto SARS, you've got 2 cities and then barely 3 and 4. And during that time, COVID spread all over China because it's just such a more exponential disease, such a more transmissive disease. And if there was another spillover 6 weeks later, I don't even know if you'd see it because it would just be lost in this noise. There's not another spillover at the same time as Wuhan. And maybe if we didn't close China, you'd eventually have another spillover. But because it was shut down, you really can't compare those two fairly.
And they're
Well, yeah, you'd also see, if you sample those cases, you could see it was different also. But they didn't sample in rural China, and then they shut down the whole country, and then they killed all the animals on their farms. So it would be hard to find, and it's a hard comparison to make between those two.
So some evidence that would speak to this is presumably there are earlier abortive transmission cases or it is likely that this is not the first time this is transmitted to humans under your...
Yeah, maybe on a farm or somewhere, and maybe if you really searched all over China, this would have been really good to do it back in zero COVID, because you could look for antibodies on those farms, and you could see, but now you can't tell because everybody in China and everybody in the world has had COVID, so you can't find that signal.
Yeah.
It would be great to look at Chinese fur farms too. They never did that. They never looked at their big raccoon dog farms in the north. There has been one fur farm in all of China in all of the world that did get infected. I wondered about that for a while. Because if raccoon dog's not the right host, it is the right host, then why don't they get infected in the wild? And I looked over all the reports, and there was one Polish raccoon dog farm in 2021 that did get infected. And then there's mink farms all over. And so you'd think a mink is a better host, but then I also found the ratio, 99% of these fur farms are mink farms in Europe, and only 1% are raccoon dog farms. So the base rate is going to be higher. So it can infect raccoon dogs, and I bet it's infected raccoon dogs on Chinese farms. They just won't tell you because China's opinion is COVID didn't start in China. I thought of a few other things we need to talk about. We didn't talk about intermediates. We didn't talk about, just going through my slides. This is just one random thing I pulled up from Yuri's tweets. Like, he's looking at the centroid and he's saying the centroid is close to the Wuhan CDC. So this is like a lab leak thing where if closeness points to something they like, it's important. But if closeness points to the market, they say that's not important. It's not really a big issue, but like
it out in retort to the zoonosis theory, because it's zoonosis makes claim its importance. So I'm just showing that it's more close to the CDC rather than But I don't think it's the CDC,
another random thing I wanted.
Truther.
Yeah, another random thing I wanted to point out is their argument really relies on a clean insertion of the furin cleavage site as being so weird. And one thing I came up with during the research is COVID has a proofreading mechanism for mutations, which influenza doesn't have. So like when you culture them, influenza mutates 23 times faster than SARS-CoV-2 in the cell culture. And I think this is probably also true in the wild. I didn't compare the exact number there. But the proofreading mechanism doesn't work for insertions and deletions. So this could be one of the reasons why everybody looks at this and says, "Oh wow, why is there a clean insertion there and no mutations on each side?" Like it filters out some of those and not the insertions. I don't know if this is the answer, but that's one thing the lab leak side might have missed.
But assuming this is true, would that explain the graph that, or how is that consistent with the graph that Yuri showed, in which observationally many of the mutations one sees in people with COVID years on, how insertions are extremely rare?
I don't know how that fits in, and I'd have to think more about that. I mean, he also showed, like, he went through the end of Banal 52 and showed, like, this is the only insertion, and why aren't there others? And so it's both the rate of mutations, but it's also the rate of evolution. So, like, the spike protein has a function, and you can't necessarily just throw big chunks into it in certain places and have it work as well. But maybe if you do throw in the cleavage site by random, then that actually worked really well. So maybe that gets favored.
Yuri, was there a reason it was only the last 1/3? Is the rest of that the ORF region? Is that the part you didn't show, or
No, just more convenient. Yeah, starting with the spike and everything towards the
it's
end.
just you didn't want to do 15 slides of
and
nucleotides?
yeah.
I appreciate that. I was sort of curious,
I'm not even sure
One of the-
needed all of the the whole genome alignment. And actually, I think Peter had a nice diagram of insertion frequency, and I think he said it's in the spike. A lot of the insertions are in the spike, but most of them in the spike are like in the first third, and there's only very few around the S1, S2 junction, like around the furin cleavage site. So it's actually, we're
in agreement. There's more in the N-terminal domain. So yeah.
And even outside the spike, even more in like the subsequent genes in that later, like after the spike, the later fifth of the genome. So just very odd to see an insertion here.
This is one other thing I want to just read into the record here. This is an interesting thing. There's this SRA is like single read archive, I think, where they put the metagenomic stuff. That's how, like, when you find the raccoon dog samples, there's sampling for COVID, but they also find animals in the reads. And so they have this archive of all these reads around the whole world. And somebody did a search early in the pandemic, like, did anyone find cryptic COVID anywhere in the world in this archive? And they couldn't find any. So that's another way we can determine that, like, it probably wasn't floating around for a long time before it showed up at the market.
So I guess he's saying they didn't find which samples? The Antarctic soil samples?
So this is just like related to the Antarctic soil samples that you're talking about. That's like one way.
Their methodology sucked.
These came in later. He looked before, he looked April 2020, and these are later. So, it's hard to prove.
Know about the Antarctic soil samples?
This is just like another Jesse Bloom thing where they like found something that looked really suspicious and it had ProCov2 cases in it, but it was actually just like late. It was like February cases that had ProCov2 which I think is a reversion. So I don't think it, like, if they could prove that these were really like November or December, it would be like a smoking gun. But, and they also had they had signs that they were actually later.
But they sequenced
samples.
it in like December 2019. So how could they have been later?
I don't think it was December. I think it was like January 30th. And it also wasn't in Wuhan. It was in like Shenzhen, I think. I think it was even the wrong
We
city.
don't have to talk about
Yeah.
Detrick.
Yeah. The only other important thing I had to talk about is the intermediates, which we haven't gotten into
Yeah,
today. Did Yuri want to go over his theory of those first, or should I just go into mine?
Go
or-
ahead. And if you have any questions, I can weigh in.
Yeah, so I mean, that's intermediates would disprove Picard's hypothesis. So those are, a lot of people point to those. If I can find my slides for those. So he ruled them out in a lot of different ways. Sorry. Oh, yeah, intermediates. So there were like 800 A or B genomes at the beginning, and then there were 20 that were CC or TT, which are the intermediates between those two. And Picard ruled them out to say it's two spillovers, and there's been a lot of speculation that this was fraudulent or, done in bad faith or whatever. And so it does seem like from my perspective, he did this correctly. Like, the work checks out. I looked into some of them. So some of these have just like super low sequencing depth at those positions, like less than 10 reads. He produced the examples
Clarify
on
what
some.
sequencing depth means.
Again, that's like where you sequence and you chop up the DNA into all these like 300 base long reads, and then you reassemble it into a genome. And then
it just means a low signal. Is that the?
Yeah, but it's a different signal across each base. So there's 30,000 bases for COVID, but you get a different amount of reads at each spot. And sometimes you get, like in these samples, we're looking at the one site and they're finding thousands of reads. And the reads are mostly, even then they're like indeterminate, but they're mostly T's. But then in the other spot, they're finding like really low signals and they're not, yeah. So you can rule some of them out that way. And then there was another case, this was in Sichuan, where they used the software that says if you didn't sequence the position, you have 0 reads for that spot. It just uses the default, which is the like Lineage B Wuhan Hu 1. So it fills in the default there. And then if you only sequence half of it, you can make an intermediate because you only sequenced half. And so Picard talked to the author there and confirmed that's what was going wrong with those. And then the other case he found was that some of these had like multiple rare mutations shared with other Lineage A's. So then you can tell that those are either like convergent evolution intermediates or misreads, because it's just extremely unlikely that they're going to share all the other mutations with those. And then there was this paper from Yuri and Alex Washburn and Stephen Massey, where they said they found a bunch of other intermediates that were like should have been included in Picard, but they like all came from Sichuan. So it's the same problem, the same software. A few were from Wuhan, but I looked those up in GISAID, and they were also, the Wuhan ones were processed in the same Sichuan university, so it's the same lab. So it's like exactly the same issue that Picard already flagged and talked to the author about, but then they wrote this paper saying, Picard's a fraud, he ignored these ones, but they're the same genomes. And this all got pointed out on the paper they wrote. There's even a funny way in which like Massey, he screwed up the font on one of these samples. It's different than the rest in his table, and it's actually a duplicate sample that included what Picard already did. So it's like, did he just change that number in the table? And hard to say. And the other funny thing is like only CC or TT can be the intermediate. It can't be both of them. But Drastic just like cites both of those to cover their bases and say it's like doubly fraudulent. That's all I had on intermediates.
Okay. Yuri, did you want to comment on intermediates at all? Or?
Sure. Yeah. Well,
If not, I have questions.
Sorry.
I mean, if you want to talk about something else, I have other questions. So.
No, I mean, yeah. In terms of
to you.
intermediates, the Wuhan genomes, I didn't realize they were sequenced in the same way as the other ones that filled in were the consensus genomes if they weren't sequenced. I have to revisit the paper, and we look at both intermediates. And either ones, like if you if you want to throw out all the CC ones, but TT ones are legit, then you still have to take into account the TT ones. That's why we looked at both, and just in terms of we were questioning the methodology of PRRAR of throwing them out, but in terms of the details of which ones are legitimately thrown out and which ones should not have been thrown out, I need to go back to the paper and kind of respond to those points of criticism. It's been a while, and some of them I didn't even see because they have those authors have me blocked on Twitter. So, but if you have other questions, feel free.
So following up on this, if I understand correctly, you're claiming that most likely lineage A is ancestral to lineage B, right?
At least phylogenetically, yeah. I mean, to think that it came from a reversion. Sorry?
I mean,
well, it could still be like B could be ancestral and A could have resulted from like double reversions to the consensus, but Peter says that's extremely unlikely, and I agree. So
so you're not. Hang on. Okay, well, I'll get to the question I was getting to, which is, I believe you talked about saying that there are cases of people who had a virus you think is ancestral even to lineage A, and therefore also ancestral to lineage B. And I just kind of wanted to understand like the context. Who are these people? Where did they get sick? When did they get sick? Did I understand what your claim was correctly?
Sure, yeah. Well, I would need to revisit all of the genomes, but I think Peter had a diagram with actual sampling dates, and basically the question goes again to, are those really ancestral genomes, or are those mutations or reversions to the kind of the ancestral variant? And Peter's claiming those are all reversions, and I think at least some of them actually do represent true ancestral genomes. And of course, if we sampled them later, I think it still doesn't mean that mutation arose later. It could be like that lineage was kind of circulating undetected, and we only sampled its kind of descendants later on. But based on the phylogeny, if you kind of don't trust the sampling dates to give you the inference of what preceded what, based on the phylogeny, we see a lot of genomes that have these mutations that are ancestral. And to think they're all reversions, I mean, that's one argument you can make, and Peter made it. I would need to revisit the exact diagrams and his analysis, because he did some of his own modeling. I mean, it's interesting to see the actual number of reversions in like both lineage A and lineage B and make that determination. But sorry, I guess I got 3 minutes left. Did I answer the question, or you had maybe some follow-up?
I mean, I'm not sure I fully understood.
Okay.
But I mean,
if I can briefly rebut, like I was saying that it's hard for this to circulate, cryptically because of the whole over-dispersed transmission, and that's why it's the single polytomy, and that's sort of true for every strain. So if PROCOV2 was the ancestor, you should have a single polytomy for PROCOV2, and it's really hard for that to, circulate. And also, sorry, just consistency of the theory too. Like this accountant Chen guy who's over near the lab, like if he was PROCOV2, like that would maybe be really interesting because it would point in multiple directions towards the lab. But he's lineage B with no mutations, so he's like downstream of the lab genetically. He's exactly equal to the strain at the lab. So either he infected the market, or straight downstream of the market. So either he infected the market or the market infected him. And I think the market infected him.
I don't know. I think unless Will has something, we might be near the end.
There's, obviously incredible density here. I think I'll have a large number of follow-up questions that I think just I have to work out what each of you have said, and then understand which questions I should ask. And I apologize, I should have done a better job beforehand, but there is a truly massive amount of information. So it's super, super cool, and it's wonderful. I just think my questions at this point will be stupid until I put pen to paper. Yeah.
Yeah, and for sure, I think it's best to answer questions in a written form, because sometimes, especially after a few hours of debating or speaking, your brain processing is going down.
We're at 7.5, so.
Oh, wow.
Yeah.
No wonder. Yes, I'm definitely feeling a bit sluggish at this point.
Okay, should we have a last, if you guys, Peter, do you want to say 20 seconds and SAR 330, or are you guys good with what's been said?
I'm good. I have a headache.
Yeah, or you took an
No,
edible.
You're welcome to answer more questions. I think there are no stupid questions, Will. I think there are stupid people, but not stupid questions.
I'll do both. Yuri?
Sure. No, I mean, yeah, I think it was very, very interesting, definitely. And I think in general, this topic is lacking in this kind of civilized debate. And I think there's people on both sides who are to blame on, dragging this to the gutter sometimes. But, I mean, I'm all for figuring out the origin, even if the origin turns out to be zoonosis and the lab leak, because the lab is not to blame, which we can all be wrong, and I was wrong several times and throughout my life, many more times, and I think we have to keep an open mind to being wrong and just examine the evidence, and have, as much as possible, to be not emotionless, like not make emotional. But it's very hard, of course, after you, especially like you've invested a lot of time into your reputation. But at least I try very hard to be objective. So if there is objective evidence that comes out through these discussions or later on, probably, I mean, because these things have been debated for so many years now that it's unlikely there'll be like a dispositive, convincing argument made on existing evidence. But maybe something will come out later on as well that will kind of be dispositive. So, I mean, with that in mind, I think existing evidence for me is more convincing of the lab leak, but I guess for Peter it's more convincing of a zoonosis. So I guess maybe it takes impartial judges that kind of haven't been exposed to all the bad actors on both sides, haven't been exposed to all of the manipulations, that can make a decision on like what at this point is the most convincing case. So in any case, I'm rambling on, and with that, thank you.
There's a lot of groupthink on both sides, and I agree that like if I'm wrong, I would like to know why I'm wrong. And I think like you're blocked by the scientists, and I'm blocked by your colleagues, and it's hard to have a civil discussion about this, and it's hard to.
And it's just,
there's no place to peer review. Like the PRRAR erratum, like somebody found a bug in his code, and it's corrected now. But like the lab leak, it's decentralized. So, where if I think parts of it are wrong, who do I complain to? So, only thing I could think of is gamble $100,000 and yell at people for 3 weeks straight.
Well, I think you've both done an exemplary job. I think there's few people who know more about it or who are committed to like producing high quality evidence. So, it's cool to see it.
yeah, and I
thank you,
I enjoyed
Yuri
it
and
very
Peter.
much, and would love to see this evidence, kind of collated to maybe, more like a written form where you can examine. I think some of the modeling that Peter did would be really interesting too.
We'll release all our slides, and I think win, lose, or draw, I think we'll make some things that people can look at and learn more about this, and
Yeah, and everybody
that'll
can
have some
draw
value,
a
and
judgment.
I don't know.
Perfect.
Okay, I'm going to stop this recording, and then we can do just logistics for next debate.
Oh, this was all on
Same
record?
time tomorrow.
Damn.